Hello, everyone, and welcome to our 2023 update on the state of real-world evidence policy. My name is Dr. Rachel Hendricks Stirrup, and I'm the Research Director of Real-World Evidence at the Duke-Margolis Center for Health Policy, housed within Duke University. The purpose of this workshop is to review Duke-Margolis's real-world evidence collaborative activities and regulatory and real-world data and evidence policy updates made over the past year. I'm proud to say we have a diverse array of speakers participating with us today for this event, working across industry, regulators as well, and individuals representing healthcare systems. I want to thank members of our Real World Evidence Collaborative as well for their contributions to our work. And I also want to thank the Margolis Family Foundation for its support, its ongoing support, in fact, of our work to date. Now I will turn to also our statement of independence. The Duke-Margolis Center for Health Policy, as I mentioned, is part of Duke University, and as such, we honor the tradition of academic independence on part of our faculty, scholars, and staff. Neither Duke nor the Margolis Center take partisan positions, but individuals are certainly free to speak their minds and express their opinions regarding important issues. Should you have anyone here have any questions about our relevant policies within our institution concerning this, you may feel free to refer to our faculty handbook, code of conduct, and other policies and procedures that are available online to anyone. Now, next, I will go over a few virtual meeting reminders for today. Attendees are encouraged to contribute throughout this entire meeting with questions using the Zoom Q&A function. This meeting is being recorded, and this recording and the slide deck will be posted on the Duke-Margolis event page in the coming days or weeks following the conclusion of this meeting today for everyone to access at their leisure. Now, here's an overview really of our agenda for today. And I see many folks are still joining, which I'm very happy about. Today, we will start with welcome and overview of real-world evidence policy updates. This will be followed by our keynote presentation, which will start at 12:50. Next, we'll provide a session 1 overview on the role of master protocols for real-world evidence. This will be followed by a break. And after the break, we'll move straight into session 2, where we will discuss evaluating real-world efficacy in patient subgroups. Lastly, for our third session, we'll go over generating practically relevant evidence in the real world. Following that, and to close out, we will have Dr. Mark McClellan, our center director, provide us with closing remarks, followed by an adjournment of today's meeting. Now, to get us started in the time that we have, I will begin with providing you all with a 2023 update on the state of real-world evidence policy. So I want to first acknowledge that today there's, and moving into the foreseeable future, in fact, there's a vast and growing amount of real-world data. There's a broad healthcare data ecosystem where there are originators, and a range of use cases, originators of data specifically, and a range of use cases in which real-world data can be accessed, used, and evaluated to answer a number of research questions and inform several operational aspects within our healthcare and health research system. There are several different stakeholders engaged throughout this entire process, including not just researchers and healthcare providers, but also contributors of and generators rather of wearables data, lab data, insurance claims data, pharmacy benefit claims data, EHR data, publicly available data through government registries, as well as an emergence of socioeconomic and behavioral data. All of this data is very important, but what we should acknowledge moving forward is that in the abundance of this data, there's an undersupply of evidence that can inform regulatory decisions, policymakers, and as I've mentioned, operations at the very fundamental level of our healthcare system. And so we are here today to see what is needed next to get us there as far as transforming all of this data into meaningful evidence that can serve stakeholders broadly, both within the United States and globally. Next slide. So this is an overview of Duke-Margolis' approach to advancing real-world evidence policy. We take a three-pronged approach specifically. We engage members of our real-world evidence collaborative. In my prior slide, we listed several members engaged in our advisory group within the real-world evidence collaborative and other key stakeholders to develop practical insights and recommendations for real-world evidence becoming a primary basis for regulatory decision-making, real-world evidence to support reasonable, equitable, and necessary coverage, and also real-world evidence to integrate research into routine care. We also take an approach to conducting research on our own, examining the ethical, legal, and social implications to real-world data implementation across the real-world data life cycle. And lastly, we offer opportunities for our undergraduate and graduate students and also postdoctoral trainees to engage with us along the way and along this journey so they are prepared to tackle next generation issues that will confront ourselves and society moving forward. Next slide. The real-world evidence, I'm proud to say, finalized its 2023 to 2027 strategic plan, so our 5-year strategic plan. As a reminder, the collaborative's mission is to drive best practices in the development, use, and application of real-world data and evidence to share learnings among industry, regulator, payer, and health system stakeholders to address their needs and also, importantly, and ultimately improve the lives of patients and drive improvements in care quality. We want to advance 4 key areas of real-world evidence policy in doing this. Real-world evidence that routinely informs decision-making, regulatory decision-making, real-world evidence that supports reasonable and necessary payer coverage, and real-world evidence that fosters collaboration and harmonization across ecosystems, including international agencies. Lastly, real-world evidence that integrates research into routine care. Next slide. Now, many different regulatory agencies, including those that you see here in the United States, Europe, the United Kingdom, China, and Canada, they all define real-world data very differently. This is a quick overview of these definitions. And one of the reasons why I'm presenting this is to show that there is work to do as far as harmonizing around not just the definition of real-world data, but also terms that operationalize best practices within the scope of real-world data implementation. Next slide. The ICMRA, or short for the International Coalitions of Medicines, Regulatory Authorities, published a statement in June 2022 highlighting this fact. One of their key areas for collaboration include harmonization of real-world data and real-world evidence terminologies, as well as convergence on real-world data and evidence guidance and best practices. Also, readiness and transparency are other important areas for collaboration. The ICMRA, or ICMRA, is comprised of regulators from Health Canada, FDA, and also the EMA. So we were proud to see this sort of joint statement that aligns with our understanding around where more work should be done to advance real-world evidence policy moving forward in the regulatory space and others. Next slide. So also in 2022, in October, a few months later from the announcement of the ICMRA statement, FDA announced its Advancing Real-World Evidence Program. This does fulfill an FDA commitment under PDUFA 7 and is incorporated as part of the FDA User Fee Reauthorization Act. What's great about this program is that it provides sponsors who are selected into the program an option or opportunity to meet with agency staff before protocol development or study initiation. Next slide. And per the FDA, the Advancing RWE program is designed to identify approaches for generating real-world data that meet regulatory requirements in support of labeling for effectiveness or meeting post-market approval study requirements. It also helps the agency process it helps the agency create processes that promote consistent decision-making and share learning regarding real-world evidence, and importantly, promotes awareness of characteristics of real-world evidence that can support regulatory decision-making. Next slide. Here's a quick timeline of what the Advancing RWE program seeks to accomplish based on information they've provided to the public. By June 2024, they will report aggregated data on an annual basis. The report will include types of data sources used, study designs employed, and regulatory requests. In December of 2025, FDA will convene a public workshop or meeting to discuss case studies focused on how to generate real-world evidence that meets regulatory requirements. And then finally, on this same day in 2026, FDA will use lessons learned from the RWE pilot program to update existing or generate new real-world evidence-related guidance documents. Next slide. I won't spend too much time on this here, but the Centers for Medicare and Medicaid Services, they've recently received an opportunity or have now an opportunity to engage in price negotiation with drug sponsors. And with that comes an opportunity for real-world evidence to play a role in this level of decision-making on behalf of CMS. Next slide. CMS has recently announced their plans for Medicare drug price negotiation. It will be interesting to see how real-world evidence plays a role in price negotiations for the drugs that they've identified as a top priority. These drugs are all listed here. And specifically, CMS or Medicare within CMS may negotiate prices directly with drug companies for these costly single source brand name Medicare Part B and Part D drugs. Next slide. If you have any questions about Duke-Margolis and our real world evidence research portfolio and engagement activities, feel free to contact me here at the information that you see on the screen. And with that, I will move to our next slide and pass it to my co-presenter with FDA, Kiel Elzarad, who's the director within the Office of Medical Policy at the Center for Drug Evaluation and Research at the FDA. Kier, please take it away.
Thank you. Thank you so much. Do you hear me well? Okay, perfect. So what I'm trying to do here is to go over a little bit of an overview, an FDA-focused overview of our real-world evidence program and the updates that hopefully will be relevant to the community. If you go to the next slide, please. Thank you. So as many of you are aware, in 2018, we published our framework. The framework is really reflective of the thinking from the Center for Drugs, Biologics, and the Oncology Center for Excellence. And it's not reflective necessarily of the Center for Drugs. Obviously, devices may warrant different considerations, and this is an acknowledgement of that. The program itself is multifaceted, and it includes here, as you see in front of you, multiple aspects, including the internal agency processes, guidance development, and demonstration project, and external engagement. If you go next, I'm touching on a few of those items today. If you want to go to the next slide. Thank you. So obviously the definitions are important, but most of us are aware of the definitions right now. It's important to highlight that real-world data and real-world evidence are different, and real-world evidence is derived from the analysis of real-world data. And this will include using study designs inclusive of randomized clinical trials. This is something that's been stressed to me over and over by an academic trialist. That's important to mention this. All three centers share the same definitions, and I think it's an important point to make. If you go next. So this paper I really like is actually from John Concado and Jacqueline Curry-Cry. John Concado, who actually the lead for the overall FDA real-world evidence program, and Jacqueline is the deputy center director. It highlights the different uses of real-world evidence, and to the very much to the left of the screen, you see the use of real-world data in the planning, which I would assume should be a best practice in general to inform site selections, accrual approaches, etc. And as you move to the right, you will see an increased use of real-world data, including on real-world data used in clinical practice, in trials in clinical practice, or point-of-care trials, as some of us may describe. If you go to the next slide. This is a chronological kind of order of our guidances that were published. And if you click next, actually, it shows the guidances that I'm going to mention a little bit. And those are from in the last few months, per se. And it shows the continuation of our efforts around guidances and around providing really the clarity needed for the community at all. If you go to the next slide. Okay, there we go. This is a small guidance, but I think it's really worth mentioning. This the guidance on submitting documents using real-world evidence and real-world data for the FDA. It's really streamlined guidance that highlight that we need to be aware of the purpose of using real-world data, real-world evidence. We need to be aware of the study design that incorporates real-world data and the real-world data sources, of course. It has a nice template in the end. And I think this is going to serve to inform us all as we move forward. You heard Rochelle mentioning the reporting aspect, but also how we can educate each other practically on how the data are being utilized and what are the sources of such data. If you go next. We also, in February, we published the consideration for the design and conduct of externally controlled trial guidance. And this is addresses multiple facets of external controls, including historical and concurrent controls. We are currently actually working on comments received in the dockets, and I want to say thank you for all of those, all of you who actually commented in this guidance. And if you go to the next slide, this is a finalization of a guidance, our guidance on the consideration of the use of real-world data. This is actually originally published in 2021, and we just finalized it as part of the mandate under 21st Century Cures. And again, thanks for the community's comment here. I think this is a cornerstone guidance for us in how we're approaching the use of real-world data and real-world evidence. It's worth mentioning. If you go to the next slide, we've listed the awards, the example of our demonstration project that we awarded. This is actually back from 2020, and the project that start producing results right now, generating results. And I just want to give some examples of them here. I'm not going to go over them in details, but I want to mention also that we are about to announce new awardees for 2023 using the same funding mechanism that you see here. So again, we really appreciate the interest from the community. As you see, we received multiple applications. And it's really excellent to see the interest and the work of the community at large to inform this area. Now, in my last few slides, if you go next, I want to touch on a little bit of an international harmonization effort here, but I know a lot of you are probably aware of ICH, the International Council for Harmonization, but I just want to make sure we're all on the same page. This council actually started by three regulators in the beginning, the US, EU, and Japan, and has expanded since to include 17 members and approximately 30 observers. So it's a very important international body that can bring us together to harmonize around critical issues. And if you go next, I want to highlight the real world aspect that's relevant here, which is, as you see in front of you, this is kind of the ICH real world reflection paper that's available for public comments right now. And actually, the fourth bullet, if you look next, is what I'm going to focus on, which is the reflection paper that's posted right now for public comment. I think the paper is available for public comment until the end of the month. If you go next, we'll touch on the content of the paper very briefly. Actually, the one before that, sorry, I think skip this slide. Thank you. This is the reflection paper, and it's really focused on the terminology and having coherent terminology and converging around understanding what do we mean when we talk about real-world data and real-world evidence in general. Hopefully, that will also lead to informing the assessment of real-world data and real-world evidence. We put the links for the paper there, and we hope this will be the start of very thoughtful approach to harmonizing around real-world, starting with the terminology. If you go next, and this is, I believe, my final slide. Also, it's worth mentioning that we're thinking of real world across the board, including in the use of real world and in the conduct of randomized clinical trials or clinical trials in general. This is an example from ICH E6R3, which is the guideline on good clinical practice (GCP). The work actually is ongoing right now. If you click next, is on Annex 2, what we call Annex 2. Annex 2 is focused on clinical trial conduct for clinical trials that will be utilizing decentralized elements, pragmatic elements, and real-world data sources. So this is not a guidance on real world per se, but it's really taking the whole ecosystem that we're seeing around us, and we're seeing how it evolved, and we're trying to address it from the point of view of the clinical trial conduct. We hope this will be useful for the community, and this will be also available publicly for comment. So we encourage you all to please stay tuned, and when this is available for public comment, to provide us with your comments. We want to make sure this is as useful as possible for the community. And then if you go to my next slide, I started listing all the people who I want to thank, and frankly, it takes 3-4 slides to list everybody. So I put the FDA here in general. It takes a village, seriously, and there's so many offices and so many great individuals who actually helped us shape this program. And this program, as you can see, is a living program. It's evolving. So a lot of the work still ahead of us. So grateful for that. I want to say special thanks for Jean Concado and his team for really being the core of this program. And again, thank you so much, and I look forward to a productive meeting. And I'll go back to you, I believe, Rochelle, right now. Thank you.
Yes, thank you so much. Well, wonderful opening. So now I'm pleased to pass it over to Dr. Donna Rivera, who will be providing our keynote presentation for today. Dr. Rivera is the Associate Director for Pharmacoepidemiology in the Oncology Center for Excellence at the FDA. Donna, happy to pass it to you.
Wonderful. Thank you so much. I just want to confirm that you can see my screen in presenter mode.
Yes.
Wonderful. Okay, great. Well, I would like to thank you so much. And I would specifically also like to thank the organizers for the opportunity to speak. I'm pleased to be here at the Duke-Margolis annual convening on the state of real-world evidence policy. So let's begin with the data. We are surrounded by data, and there's certainly an increasing amount of available real-world data. The question is how or if that data can become real-world evidence. And I think the beginning of that question is very important because it gets at the core. What is underneath all of this data? Well, it is the patients, people, information about individual people and their care, and how it is conveyed through underlying data. And this is fundamental to how the promise of real-world data can bring greater patient centricity and generalizability to evidence generation. To learn more about how patient outcomes are really displayed in the real world is how to connect data with people and understanding this. And from a regulatory perspective, there is a need to deeply understand how the data were collected, if there's an availability for auditing, an ability to understand uncertainty and be able to rely on the data generated. This is to ensure when data and evidence are submitted for medical product review, regulatory standards might be met for safety and effectiveness to evaluate patient benefit and benefit risk within the framework. So similarly to clinical trials, what this means is we need to remove uncertainty and bring clarity. We need to understand the data, the patient, the assessments, the source quality, which means missingness, auditing, ability to verify and assess data relevance and reliability. To achieve patient-centric evidence generation and reflect what is happening in the real world, to do this requires this understanding to improve the ability for generalizability and representative real-world evidence. So let's focus on how we are trying to accomplish this through the FDA Oncology Real-World Evidence Program. The FDA Oncology Real-World Evidence Program aims to collaboratively advance the appropriate use of real-world evidence in oncology product development to facilitate patient-centered regulatory decision-making. The program was established with 4 strategic priorities around optimizing knowledge building through a centralized oncology real data resource portfolio that ensures study efficiency, transparency, and diversity, advancing the scientific development of resources, regulatory policy, and guidance, collaborating through strategic partnerships and collaborations across the agency, as well as through external organizations, and to accelerate the field of oncology RWE through leadership and training in rigorous evaluation, methods development, and regulatory science. The Oncology RWE program was established in 2020, and I'm going to provide a current overview of where we are in year 3. The Oncology RWE program has 4 focus areas: regulatory review, regulatory science research, collaboration and outreach, and education and engagement. In the area of regulatory review, our team has completed around 200 reviews across CBER, CDER, and CDRH. as well as participating in various RWE guidances led by the Office of Medical Policy. In the regulatory science research category, we have various examples of research throughout a variety of mechanisms, which I'll discuss in just a moment. For collaborations and outreach, we are actively engaged in efforts with the White House Cancer Moonshot, as well as engaged in a variety of publications, presentations, and posters presented within the scientific community. In the area of education and engagement, we are invested in education and training around the use of real-world data through efforts such as Project Socrates, various fellowship programs, specific curriculum, academic teaching, and Team Forward. Team Forward, or translational evaluation and assessment of methods to facilitate use of oncology real-world data, Team Forward, was originally formed because there's no better way to learn about real-world data than to review real-world data in a regulatory context. So this group is comprised of active reviewers with epidemiology, clinical, statistical, and regulatory science expertise to bring awareness of real-world data across drugs, biologics, and devices. This team is intended to bring in another layer of familiarity around the appropriate use of real-world data in regulatory review. And not just reviewing what's been done, but what could be done in drug development plans and new opportunities for use of real-world data in regulatory contexts to complement our understanding of risks and benefits for patients. This team also highlights the collaborative and interdisciplinary nature necessary for review of real-world data applications, bringing together clinical, biostatistical, and epidemiological knowledge through a patient-centric approach. And since regulatory review is at the center of what we do, I'm going to provide a brief overview of what we are doing in this context, as it is part of our central mission as an agency. Real-world data and real-world evidence can be used for various regulatory purposes, including to support the effectiveness and/or safety of a new molecular entity or unapproved product, to support labeling changes for an approved product, to support or satisfy a post-marketing requirement or post-marketing commitment, or as contextual or supportive evidence. When using real-world data for any of these purposes, and in all study designs, when using real-world data, there are many factors to consider, and I want to highlight the importance of two: reliability, including data accuracy, completeness, provenance, and traceability, and relevance, including the availability of key data elements and sufficient numbers of representative patients, which contribute to a data source being fit for purpose for a regulatory objective. The strength of real-world evidence submitted in support of a regulatory decision depends on the clinical study design, methodology, and the reliability and relevance of the underlying data source. After rigorous evaluation, there have been a few examples in FDA oncology of the use of real-world data to support approvals, which I would like to discuss now. Alpelisib received accelerated approval in April of 2022 for the treatment of patients 2 years of age or older with severe manifestations of PIK3CA-related overgrowth spectrum who require systemic therapy. Based on data from EPIC-P1, a single-arm clinical study in patients treated in an expanded access program for compassionate use enrolled across 7 sites in 5 countries, there was data from medical charts and radiographic images at participating clinical sites. In the context of this application, it was unfeasible to do a clinical trial. And in a rare disease with a high unmet medical need, there were no approved therapies available. This particular submission had study objectives and endpoints, a statistical analysis plan, and enrollment guidance that were predefined, with objective outcome validation, including images that were available with blinded independent central review, as well as other data on functional improvement. As regulatory context, the conduct of a single-arm study and the use of data with BICR from the Expanded Access Program was considered appropriate given the rarity of PROS and high unmet medical need. The results of EPIC-P1 provide substantial evidence of effectiveness, demonstrating clinically meaningful and highly durable responses as assessed by BICR. to provide substantial evidence of effectiveness and a positive benefit risk assessment. And the second example is abatacept. In December of 2021, abatacept, a selective T-cell co-stimulation modulator, was approved for the prophylaxis of acute graft versus host disease in combination with a calcineurin inhibitor and methotrexate in adult and pediatric patients 2 years of age and older undergoing hematopoietic stem cell transplantation from a matched or one allele mismatch unrelated donor. This is the first drug approved to prevent acute graft versus host disease. Safety and efficacy was evaluated in 2 studies, GVHD1, a randomized controlled trial in 8 out of 8 HLA matched patients, and GVHD2, a clinical study in 7 out of 8 patients. I'm going to focus on GVHD2 and the data source for that, which was the Center for International Blood and Marrow Transplant Research, CIBMTR registry. This registry includes data on every allogeneic transplantation performed in the United States as required by law. The data source provides comprehensive information, including demographic, clinical, and transplant characteristics. Together, these studies resulted in approval of abatacept for the treatment of acute graft versus host disease. So based on these examples and current guidance, the use of real-world data for regulatory purposes should include careful planning to think about predefining all essential elements of a study, including design, analysis, and conduct, to describe how each element was ascertained from the data source, to address issues essential to determining reliability and relevance of the data in the protocol, including ability for auditing, and to consult with the FDA and relevant review division early and often prior to the start of study on study details. Much of the information discussed within these recent slides is represented in the newest RWE guidances. As the use of RWE increases, the FDA is responsive to drug development needs and has released various guidances as mentioned and led by the Office of Medical Policy in collaboration with CDER, CBER, and OCE. These guidances include information on real-world data sources, such as electronic health records, medical claims and registries, on real-world data submissions, and on study design, specifically the most recently released guidance as it pertains to externally controlled trials. For additional details, please consult the guidances directly. So as we think a bit about how our work is centered around regulatory review, the opportunity to collaboratively conduct regulatory science research affords us an opportunity to advance regulatory science research, which enhances regulatory review. In this synergistic approach, science informs review and review informs science. So with this background, I'm excited to talk about my perspective on the current state of oncology RWE by focusing on where we are and where we are going in 2 areas, innovative science and innovative design. Starting with innovative science, the Oncology Center of Excellence has a scientific collaborative that focuses on enhancing regulatory science research in oncology. The OCE has 9 scientific interest areas and 1 cross-cutting area, real-world data, for its applied research efforts. Because of that, in real-world data, we work across many of the OCE projects and programs, which are highlighted here, and focus on various opportunities for topics, methods, and mechanisms to advance oncology drug development for patients. Specifically in real-world evidence, our program was established with 4 initial goals in the context of research. Those are to foster consistent terminology, enhance source data, standardize regulatory submissions, and develop real-world endpoints. We decided on these initial goals as a practical approach to lead to tangible accomplishments. So within OCE and across FDA, there are various mechanisms to collaborate with FDA for regulatory science research. Regulatory science research is defined as science of developing tools, standards, and approaches to assess the safety, efficacy, quality, and performance of FDA-regulated products. And these pillars of regulatory science represent the goals of modernizing and developing evaluation of regulated products, strengthening post-market surveillance and labeling, and invigorating public health response. So within this, there are many ways to collaborate with the FDA through CIRCs, grants, RCAs, partnerships, and BAAs. I'm going to walk through each of these individually and present a handful of examples that reflect the broader oncology research portfolio. So in the context of the CIRCs, or Centers for Excellence in Regulatory Science and Innovation, this is representative of the University of Maryland CIRC project. Building capacity for research on cancer and aging, the University of Maryland Greenbaum Comprehensive Cancer Center Medicare database. The objective of this project is to evaluate the feasibility of creating database infrastructure to support translational research and identify determinants of disparities in cancer outcomes, particularly among underrepresented and older adult populations. Uniquely, the University of Maryland Greenbaum Comprehensive Cancer Center serves a diverse population that encapsulates the ability to analyze and understand underrepresented populations. And this is done through a linkage with cancer registry data, Medicare claims data, and electronic health records. This database will offer opportunities to advance research capacity to assess multi-level determinants of cancer outcomes in pharmacoepidemiology and oncology health services research. Next is research collaboration agreements. The Oncology Center of Excellence has collaborative agreements with a variety of real-world data groups through our scientific collaborative, providing the opportunity for direct evaluation and analysis of oncology real-world data. Presented here are posters and publications from professional meetings covering research across therapeutic areas on topics such as methods and advancing health equity. And these publications reflect efforts to increase understanding of available real-world data in oncology from collaborative research studies with groups such as CODA, Flatiron, and SciAps. Next, we will talk about cooperative grants. I will highlight the Oncology Q Card, a project in collaboration with the Reagan-Udall Foundation for the FDA. The Oncology Quality Characterization and Assessment of Real-World Data was a project brought on by regulatory need. The oncology review divisions were receiving a high volume of real-world data containing submissions in a non-standardized format. And these initial submissions provided insufficient information for substantive review, requiring the continuous need for clarification. Therefore, the objective of this project was to provide a structure that can support the inclusion of sufficient information for this initial characterization to increase efficiencies and facilitate more effective communication. From an external partnerships perspective, I would like to highlight the Friends of Cancer Research Collaborative Real-World Response Pilot, a unique multi-stakeholder partnership built on a common protocol and statistical analysis plan, with the objectives of assessing the availability and frequency of core data components for measuring real-world response, including imaging and clinician assessment, as well as the ability to evaluate the consistency of real-world response outcome measures across data sources in an aligned patient population. I would also like to highlight the Cancer Moonshot, focused on delivering innovation to patients and communities with objectives such as creating a learning healthcare system and designing clinical trials to be more streamlined and accessible to all patients. The work being done is through the Cancer Cabinet, which represents various federal agencies and departments. The Cancer Cabinet also has a focus on data and innovation with the following themes of data interoperability, clinical trial innovation, and artificial intelligence. And finally, I'd like to discuss examples of broad agency announcements. This particular collaboration with the University of Oklahoma is on analyzing outcomes and genomic data of American Indian patient populations treated with immune checkpoint inhibitors. Because of the knowledge gap and the lack of current studies examining this population receiving immune checkpoint inhibitor treatment, The study aimed to develop a multi-center national database that could report on clinical outcomes and toxicity profiles while evaluating the tumor microenvironment and biomarkers. And this, presented at ASCO, is a poster that shows the early results and availability of creating such a database to better understand outcomes in the American Indian patient population. I'd also like to mention a newly funded study, ENCORE. ENCORE is the emulation of interventional trials in clinical oncology with real-world evidence. ENCORE builds on the foundation of the OMP-funded RCT duplicate project, which aimed to use trial emulation approaches to develop methodological and data insights. And through this project, ENCORE will demonstrate and empirically test the process through evaluating 12 oncology trials across 4 cancers through a systematic process. And this project is ongoing. So bringing this all together and revisiting our initial goals, I'd like to talk about our progress. In the context of enhancing source data, we discussed the BAA and CIRCE projects. We also have ongoing work with Alliance and MCODE, as well as the ASH Collaborative, to think about how there are ways to evaluate and enhance data at the source, as the underlying quality of the data is paramount. And right now we have established collaborations that are ongoing. From a standardizing submissions perspective, the Oncology Q Card collaboration with the Reagan-Udall Foundation worked to do just that, and the publication is pending. And there's a lot of discussion around the use of real-world endpoints development through the real-world response study with the Friends of Cancer Research, where there was just a recent public meeting and the publication is pending. More broadly, I'd also like to thank Duke-Margolis for their efforts, including the RWE Collaborative, where the FDA can observe ongoing efforts within the RWE community. And as highlighted in this meeting, efforts on master protocols and understanding patient subgroups using real-world data. Meetings such as this are important for increasing transparency, communication, and understanding. So now I'd like to shift to talking about the other part of the State of Oncology RWE, innovative design. Presented here is a model for clinical evidence generation, within clinical trial data being on the far left and real-world data being on the far right. Data can be generated from clinical trials and observational studies to comprise clinical evidence. And it's important to think about how we can incorporate various types of evidence to be most effective and patient-centric. This is notably shown as a continuum and not a dichotomy. And as shown are how different designs fall within this continuum. So on the far left, traditional clinical trials are prospective, interventional, with the ability to randomize, systematic assessment frequency, highly monitor protocol-based care, and have strict eligibility criteria typically. As we move to the right, decentralized or hybrid trials may allow for flexibilities in assessment and have the ability for remote or virtual arms. Moving further to the right with pragmatic trials, there's an opportunity for embedded assessments with selective monitoring, affording broader population eligibility. And finally, on the far right, traditional observational studies are often retrospective, non-interventional, non-randomized, with routine clinical assessments in routine clinical care, reflecting the broadest population eligibility. And so we think about the current state and how we approach traditional clinical trials are often complex and restrictive with resource-intensive needs and can be highly burdensome for patients and providers. So I'd like to envision a future state where there is a possibility, when feasible and appropriate, to have more streamlined and generalizable target, objective-focused approaches that are closer to routine care, affording increased access within communities and appropriate flexibility. And directly to this point, the Oncology Center of Excellence has recently launched Project Pragmatica, focused on advancing evidence generation for approved medical products by exploring innovative trial design approaches that introduce functional efficiencies and patient centricity through integration with real-world routine clinical practice. And building on this and thinking about the potential for various pragmatic models and introduction of pragmatic elements, the elements of eligibility, recruitment, setting, organization, flexibilities, follow-up, and outcome and analysis, as we think about how each of these elements may be optimized to bring trials to more streamlined approaches and allow them to be more patient-centric. And again, all of these elements, when individually designed, should be thought through carefully and evaluated for the possibility to introduce such elements where necessary or appropriate into different clinical trial approaches. And doing so is really moving clinical trials closer to reflecting the real world population by bringing trials to patients and utilizing novel approaches while maintaining data integrity. There's an opportunity to enhance generalizability and understand routine clinical practice. And there is much to be done to get closer to that goal. So I'd like to conclude by talking about just that, possibilities and challenges. So there are a lot of possibilities in the context of real world data in oncology and beyond. Thinking about the rapid characterization of emergent public health needs as shown by COVID-19, innovative prospective designs in the use of pragmatic trials, for example, real world response data assessment, ascertainment, and validation, understanding drug effects in underrepresented populations. And then there are challenges to source data and quality, the ability to make causal inferences in non-interventional studies, and the availability and analysis of raw imaging data for response endpoints. And while these do represent challenges, I think there's a lot of progress, and there are certainly more possibilities than there are challenges, and a lot of collective work for our community together. So in thinking about where we are in the state of oncology RWE, there is thankfully a brilliant amount of innovative science and a lot of thought towards the possibilities and potential for the future of innovative design. And to that, how we can envision the future and where we can have the ability to bring clinical practice and clinical research closer together at the point of care so that we are allowed and able to truly generate reflective real-world evidence from real-world data that maintains data integrity, study integrity, can be relied upon, and also allows this opportunity for the bridging of these aspects to truly represent patient-centric evidence generation. So with that, I would like to acknowledge my wonderful colleagues, especially the mentorship of Dr. Paul Klutz in building this program, as well as OCE leadership, colleagues across various centers and offices, and all of the work that we're doing together collaboratively across the agency to advance the use of real-world data. Thank you so much, and I will turn it back to the organizers.
Thank you so much. What a wonderful keynote talk, Donna. We are always pleased to have you with us, and we're very grateful for the very robust and informative overview that you've given us today. We will go ahead and get started and moving into our first session of the day. I do want to first and foremost remind our audience, should you have any questions, please feel free to use the Zoom Q&A feature. Again, if you have any questions for our speakers and panelists that have already spoken today or that will speak throughout the remainder of the day as we move into our sessions, please feel free to use the Q&A feature. Don't be shy. This is a public forum, and we would love to have everyone's engagement should any questions come up from our audience members. So that being said, I'll go ahead and move into session 1, where we'll discuss the role of master protocols for real-world evidence. In this session, real-world evidence master protocols will be discussed, specifically focusing on how to draw from clinical trials master and common protocol concepts. Our panelists will consider how different research contexts can use master protocols to fit research questions that are answerable through leveraging real-world evidence, as well as considerations for designing prospective and retrospective studies using these kinds of protocols. Before I hand it over to my panelists, I'm going to provide a short overview of our real-world evidence master protocols white paper that was released today as an advanced copy. It is published on our website, and we will provide a link to this paper in the chat for those of you who are interested. I encourage everyone to read it when you get a chance. But in the meantime, we'll go ahead and kick off with me providing an overview of what you can expect to read in the white paper. So first, I'll start with a quick policy overview around why this topic is important. So as you've learned so far, the FDA's real-world data and evidence evaluation criteria concern or apply to a data's relevance for a product's proposed indication. With that, they expect clear and well-assessed outcomes and methods used that minimize bias, as well as rigorous statistical analysis. So past FDA master protocol-related work includes FDA's Sentinel initiatives, COVID-19 natural history study, and protocols for distributed queries. The FDA OCE, in fact, had a CODA partnership entitled A Master Protocol for Understanding the Applications of Real-World Evidence in Oncology. The Reagan-Udall Foundation, I'm happy to say, for the FDA, released a COVID-19 evidence accelerator that also embedded master protocol elements therein. In March 2022, guidance was released on master protocols, and in August 2023, guidance was released on real-world evidence considerations. All of these items here can in more or less ways tie back to what we are going to discuss today as far as building real-world evidence master protocols and key considerations in operationalizing the protocols, as well as latest developments in the scientific literature. Next slide. So I'm going to start with giving you an overview of our work stream. So our master protocols work stream lived throughout our 2022 Real World Evidence Collaborative membership year. The white paper that essentially highlights activities within this group is what was published today. Again, I'm proud to say. It's important to note that master protocols have, for a while now, supported traditional randomized controlled clinical trials across multiple sites, essentially allowing multiple sites to conduct studies in a way that is in a way that converges their operations, again, to a singular research question, oftentimes by a single study sponsor. The protocols allow for multiple researchers multiple researchers across multiple sites to test multiple research questions that, again, converge around a single important research question regarding different treatments and patient populations. So, for example, thinking of basket trials, umbrella trials, platform trials, et cetera. And what's helpful is that these kinds of studies help improve study efficiency, scale, and consistency, again, across a range of different sites and questions that are answerable, that align to a specific research question of interest to a sponsor. But not all lessons from randomized control trial master protocols are applicable for real-world evidence studies, but they do provide a foundation, and that's why we want to discuss this today and disseminate our findings from our white paper. Next slide. So here is a quick overview as far as opportunities to develop real-world evidence master protocols. Again, thinking about real-world evidence studies, master protocols to support them in ways that build on what already exists within the clinical trial space as far as operationalizing master protocols, but adapting them to the real-world evidence research landscape. Developing real-world evidence master protocols to answer key research questions involves gathering evidence, for example, about what we've learned, COVID-19 therapeutics in real time. So identifying common data elements, it starts there. Harmonizing common data element definitions is the next step. Thereafter, developing standardized study design and analysis plans, and then lastly, combining their results. These are the 4 critical steps through which real-world evidence master protocol can be helpful to sponsors and regulators. Next slide. So as I've mentioned, master protocols are a well-established method for efficiently and systematically testing multiple hypotheses within a single clinical trial. We explored how these methods could apply to real-world data and evidence studies. We built upon prior multi-stakeholder efforts to generate observational evidence, or rather master protocols can facilitate this And we aim to evaluate how a real-world evidence master protocol framework and principles can be refined to meet the needs of the public. And here's an image of our advanced copy that you all can access through the link in the chat. Next slide. So I'll move into our white paper overview before we move into the panel discussion. So this white paper was informed, as I've mentioned, by our 2022 Real-World Evidence Collaborative Workstream. We hosted a virtual private workshop on February 28th and March 1st on the following topics: key terms and definitions and concepts for the development and implementation of master protocols, engaging stakeholders around the value of master protocols to support real-world evidence studies, as well as data requirements for retrospective and prospective real-world data studies. Next slide. So starting with shared terminology, definitions, and concepts. We thought, how should participating stakeholders define their real-world evidence master protocol and their key components? It's important to first, again, as I've mentioned, align on inclusion and exclusion criteria, common data elements, operational definitions, data curation and analysis plans, etc., and distinguish between wanted versus unwanted variation. Ideally, a master protocol should streamline diverse real-world data sources across multiple study sites and designs to balance trade-offs between precision and accuracy versus study efficiency. In my next slide, we sort of provide a nice sort of disambiguation to illustrate this. So ideally, we want to strive towards high precision and accuracy, and in addition to that, high study efficiency. And this is typically where we see or would find trials with pragmatic elements in clinical practice settings. And this is where master protocols have the highest potential to be of the greatest utility to researchers, including sponsors and regulators that have an interest, again, in addressing multiple research questions or hypotheses within a specific clinical trial setting. Next slide. Some source data considerations are that data quality is paramount in any real-world evidence study discussion. Developing high-quality data sets can be a multi-step process that will vary by according to research context. That includes the research question, the resources that are available, lending to whether or not a study is feasible within a given timeline, as well as the stakeholders that must be involved along the way. And different contexts necessitate different data quality considerations as well. So for example, regulatory decision-making studies to support that versus natural history studies that allow researchers to understand the natural progression or etiology of a given disease or disorder. Next slide. Other source data considerations are at minimum, a minimum necessary standard for data quality provides clarity among stakeholders for executing a useful or what could be or should be a useful real-world evidence master protocol. So thinking about common protocol designs, data models, programming, bias control, validation, et cetera. Also, with determining data, whether or not it's fit for purpose, so whether or not it's relevant and reliable. And also, I will add there, of sufficient quality. Tools like Harper and SPIF2 can support harmonization. And we dive into more detail around this within our white paper, again, drawing from lessons learned within the literature as far as Harper and SPIF2. This is something that you can expect to read in our advanced copy. Common data models are always helpful, but they're not always necessary within a real-world evidence master protocol. And again, we dive more into those details in our white paper, and we'll certainly talk more about that today with our panelists. Next slide. Now, within, again, source data considerations, it's important to choose protocol tools and design protocol infrastructure to minimize bias and control for confounding factors. That includes scrutinizing data sets for accuracy, completeness, and missingness. Provenance, completeness, and missingness is very important, especially if imputational methods are considered where necessary to address missing data for a small subgroup or subset of data subjects. Stakeholder communication and consensus is always required at every step to ensure everyone is operating along the same pace and along the same guidelines. Timing is especially relevant, particularly or especially with secondary uses of real-world data sources like EHRs or claims data. and understanding time horizons and windows of exposure, data latency. These are all very important source data considerations that are, again, addressed in our white paper. Next slide. Factors towards developing and leveraging real-world evidence master protocols. Real-world evidence is inherently challenged with substantial variation regarding real-world data capture, curation, and transformation into a broad data set. And that's why data requirements are so important. Though it's impractical to set a universal data requirement or standard across an indefinite breadth of real-world evidence research, there is, for that reason, going to be some sort of acceptable level of some unwanted variation to expect. And again, it's up to investigators to decide what is acceptable versus not acceptable. And therefore, it's important to align stakeholders in this process on the degree of master or commonality of greatest return on investment following the study implementation. Next slide. Supporting stakeholder engagement. We will constantly recommend this through and through. It's important to incorporate diverse stakeholder perspectives into the protocol development phase at the earliest phase all the way through execution, engaging stakeholders through value proposition, or at least providing that to them to encourage or sustain their participation. And lastly, involving patients, sponsors, regulators, health systems, and providers, real-world evidence policy stakeholders generally in the master protocol design process is very important in order to ensure that, again, all of the parameters are set around wanted versus unwanted variation in the results. And so also that everyone is aware of all of the operational aspects that they must undertake in order to ensure study success in leveraging a real-world evidence master protocol. Next slide. Now for our key takeaways before we move into our panel discussion, they are what you see here on the screen. Real-world evidence master protocols aim to improve consistency and efficiency in real-world data and evidence studies, leveraging, as it is practical, the standardization mechanisms that are inherent to master protocols may reduce variation and produce more reliable, comparable real-world evidence. In reducing variation, it also allows for set parameters and expectations around, acceptable levels of unwanted variation as well. And biomedical innovation and real-world evidence credibility will advance when multi-stakeholder-led actions coordinate shared resources to address evidentiary gaps. Again, another value add that real-world evidence master protocols can offer. Next slide. We'll move into our panel discussion. Now I'm happy to introduce our panelists. I'll be moderating this discussion, and I'll ask all of my panelists to now, if you're able, please come on camera and unmute yourselves. I'm really happy to have all of you here with us today. It's my pleasure to introduce each of you. Here we have with us Mark Berger, who's a semi-retired part-time consultant and scientific advisor concerning all things real-world evidence. We also have Elise Berliner, who is the Global Senior Principal for Real-World Evidence Strategy at Cerner and Visa, an Oracle company. Next, we have Jeff Brown, who is the Chief Scientific Officer at TriNetX and also a part-time lecturer at Harvard Medical School. And we also have Josh Vessel, who is the Senior Clinical Advisor at the NCATS or ACATS's Division of Clinical Innovation. Now, next, what I'm going to do is pass it to my panelists, who will each provide a few minutes of remarks that they'd like to share with everyone as we move into our Q&A discussion after. I do have some questions for our panelists today. And again, I encourage everyone in our audience to also submit questions for our panelists or for me using the Q&A function that's available through our Zoom platform. That being said, I'm going to go ahead and pass it first to Mark Berger, who will have a few minutes to give us to share his opening remarks for our session 1 discussion.
Oh, thanks, Rochelle. I think the overview talks we've had today from Rachel and from our keynote speaker have really framed the discussion very well. As we move to try and bring the clinical practice of medicine and the research, medical research together to create a learning healthcare system, there are challenges, and the challenges have to do with things that have been discussed, which is that there is sources of unwanted variation, which will confound the ability for research studies to detect true treatment effects. And by using a master protocol, one can hope to minimize that. You can't eliminate it entirely, but you can hope to minimize it. On the other hand, the other thing it can do is if one can use some central infrastructure, you can take some of the burden off of individual sites in terms of executing those studies. And that will lower the burden on individual investigators from participating, and also makes the overall cost of the study lower. With regard to that, one of the biggest challenges is that if we apply the same good clinical practice guidelines to pragmatic or point of care studies that use master protocols that we do for standard clinical research, it's going to represent still a huge barrier for expanding the population of investigators who want to participate in research. Of course, we want to now get to investigators who have not been part of the clinical research enterprise and have not been having an infrastructure and assistance helping them running clinical trials. And so we're going to have to think about what does it mean to be an investigator, and how do we incorporate the execution of the study into the general flow of the actual practices that we're trying to recruit. Because if it's not integrated into their flow, then it's going to be a huge burden on them and they will not participate. I'll stop there.
Thank you, Mark. Next, I will turn it to Elise Berliner, who will provide her opening remarks for the panel.
Thank you so much for having me here. Just to underscore some of the things that have been said already, lots of studies are published every year, but it's difficult or impossible to synthesize and compare because of differences in methodology. And we want to really reach a place where we can compare apples to apples, for example, being able to create external control arms. Some of the major definitions that need to be harmonized include inclusion and exclusion criteria and outcome definitions. And I just also want to point out that this is also a problem in clinical trials, that different clinical trials use different definitions, making it really hard to compare and understand the results across trials. And there are efforts such as ICHOM and the COMET program in the UK to try to do outcome measure harmonization and other kinds of harmonization. But even when you've harmonized those major definitions, there's still minor differences in implementation that could occur. So, for example, the Operan project is looking at how different groups are, even when they have a common study design, what are the differences in how they're operationalizing it? And they've published a study showing that they actually, using even the same data, the same goals, they've come to different cohorts because of small differences or seemingly small differences in how they've operationalized definitions. And so I just want to make 3 points about maybe ways to go in the future. So one is, I think that we should really think about the role of registries. So, we kind of forget registries a lot, but registries have advantages in that they have common data dictionaries, and observational studies and RCTs can be nested in a registry infrastructure. And in some cases, that has been really shown to be an efficient way to collect data for multiple purposes and harmonize data across clinical sites. The other thing is that, another point is that, efforts to create master protocols for real world evidence are tied to harmonization and interoperability efforts in general. So, we do really have an issue with standardization and interoperability of healthcare data, as you all know. So there are many different groups working on this problem. And so I'll just point out one, the SHIELD project, which has the goal to standardize coding for laboratory tests. So their goal is to describe the same test the same way every time. And they published a paper looking at 331 tests available in laboratory catalogs. 136 or 41% were represented by mismatched LOINC codes across 5 medical centers. So, you know, I mean, we really have to solve those basic problems. And I know it seems really impossible to solve them, but we're all stakeholders in the healthcare system, and we can't talk about what we're doing in a vacuum separate from all these other efforts. And then the final point is that you know, there's increasingly huge amounts of buzz about machine learning and computable phenotypes for defining patient cohorts and outcomes. And even more than that, now people are talking about using machine learning for the documentation of real world evidence in the or clinical care in the EHRs. So, you know, these methods might overcome issues with coding, but how does that fit into all of these ideas of master protocols and harmonization across sites? So I think that that is something that really needs to be studied more. So those are my comments.
Wonderful. Thank you so much. I'm already getting excited about what's to come for this discussion, but next I'll turn it to Jeff. Jeff, please take it away with your opening remarks.
Great, thanks a lot. And thanks to Duke-Margolis for teeing up such great topics. Just really consistently hitting topics that I think just, they need to be discussed. And it's, to me, the answers aren't that clear. In a way, I feel like I'm coming full circle. I'll admit I spent most of my career avoiding master protocols and building systems to avoid them on purpose because they're so difficult. So reality is there's a long history of master protocols in real world data, especially before common data models, right? So before common data models, you had to write a protocol, send the protocol to a bunch of our partners and hope that they all implemented them identically, hopefully. It never happened. You can't ever really implement them identically. It's just the way it goes, which is why I ended up that system started to build on common data models, Cornet, Sentinel, others. Were all designed to try to avoid it. I'll give one simple example. I do have a positive spin, I promise, because I think there's a good place to go. But the simple example was the protocol said exclude all patients with a diagnosis of cancer. This is the ICD-9 field space. So someone looked and said, okay, well, ICD-9 X to Y, that's all cancer, excluded them. Someone else looked at that and said, you would never exclude basal cell carcinoma from this cohort, and didn't. So, of course, no one ever told us what they did. So we ended up with cohorts that didn't quite look alike. And why was that? And two people, they just implemented differently. There's a bunch of examples actually in that same study where thoughtful people made different decisions because the master protocol wasn't specific enough. So I think that's what we learned, is all those lessons are leading to what was described earlier, like the paper and all the real world stuff, evidence work that Margolis has been doing to try to come up with better ways of writing master protocols. And it comes down to things like specifications. You can't just say exclude patients with cancer. You need a code list. You can't just use NDC codes. You need RxNorm codes, right? You have to actually get to that level of specificity. The really simple example is age as of when. It's really easy to say stratify by age, but in a 10-year study, if you don't say when, people do it wrong. Not wrong, differently, right? So those are, it seems simple, but the level of specificity is so important. So I wanted to kind of end with the scenarios. I do think we have to think through when master protocols are really needed and when they're not. It can't always be needed. And what we're trying to do, what is the goal? In my view, the best chance of success, and no one's gonna, I'll put it out there, the best chance of success that I think is something that's tightly coupled, meaning a group decides they really want to generate evidence across a range of data sources or partners or whatever. And there's a single funder and there's a single team, and they work together to make it right. And they actually talk every week. That's what I would call a tightly coupled approach. The other extreme is someone writes a protocol and hopes someone else anywhere else in the world can implement. I think we're probably unlikely to get what we think we're getting, is maybe the easiest way to think about it. So those are two really different scenarios. And you can imagine regulators working closely together to study a safety question in Europe and Asia and the US. If they do that together, pretty good chance of getting robust information. If they don't work together on it and expect the protocol to drive, I'm less confident of how we'd be able to interpret results there. But this is a really important question, because there are topics that require broader collaboration than we have right now, and master protocols is a way to try to drive that. So we have to kind of work it out. I'll leave it there.
Yeah, thank you, Jeff, so much. And thank you again, Mark, Elise, and Jeff, for your remarks so far. Last, I'll turn it to Josh. Josh, go right ahead.
Hey, thanks so much. Thanks, everybody. And I really appreciate the opportunity to be here. This is a great conversation. Going last, probably the smartest thing that I could say at this point is, yeah, what they said. Because you've already heard some great stuff here, but I'll pick up a couple of threads that have stuck with me throughout the meeting so far. I love, first of all, I really enjoyed reading the draft of the white paper, and I really appreciate your overview of it. The concept of unwanted versus wanted variation, I think, is a really nifty one to grab a hold of, because I think that, and it gets to a broader theme of why I love these discussions about real world data and real world evidence, is that when we talk about this and really try to get our brains around the full breadth of it, it forces us to challenge our assumptions about how we think we can know things. And I think the wanted versus unwanted variation is a really important one, because a good example of it, because I think for a lot of us, the idea that, well, variation is something to be avoided and stamped out, and but that's how we determine, as Mark said, whether there's a treatment effect or not. And it's where is that variation? Another theme that I've heard come out that I think is really important to pick up on and highlight is the idea of, and I'll confess, I don't love the phrase fit for purpose, but the idea of taking a step back and saying, what are you trying to know and how do you need to know it? Jeff, you mentioned this a little bit. Do you need real world data and real world evidence for the question you're trying to answer? Sometimes yes, sometimes no. Do you need a master protocol to engage with that real world data and convert it to real world evidence? Sometimes probably yes, maybe sometimes no. And so addressing these very basic questions in a very systematic way, I think can help us think through these, recognize assumptions where they exist, and make sure that the assumptions apply, just like you would if you were trying to decide, is a real world data set of whatever description appropriate to the question you're asking? Is it in fact fit for purpose, if we're going to use the phrase? And then the other thing that I think is really important to highlight, and this is, for me, this has come up in a whole bunch of different discussions that I've had, is the idea not only of training, which I think we heard a bit about from Dr. Rivera in her outstanding talk, but the idea of retraining. I've tried to develop a synapse in my brain that fires immediately when we talk about training in this space to say the word retraining as well, because there are a lot of people who are engaged in the clinical research enterprise that don't have enough experience or don't maybe have the right experience to come into this in the way that they want to. And Mark, I think you touched on this a bit of the idea that if we do this right in terms of constructing things like master protocols that get us all singing from the same songbook, it does have this effect of being an invitation, an inclusive invitation to bring investigators in. And as you were talking, I thought you know, as part of this training and retraining idea that I think is going to be an important maybe side benefit, maybe main benefit of these kinds of discussions. You know, every good master protocol, in addition to producing high quality evidence that decisions can be made from, it also becomes an important playbook for how somebody else might do something similar. And that's a huge benefit, especially I think in this space. We all sort of take that as a given that yes, you know, you look at existing protocols and you publish your protocols, and there are ways to do that even before the study is under full sail for traditional clinical trials. But I think it's even more important in the real world data and real world evidence space. So I think I'll leave it at that because I know we have a lot of other stuff to talk about, but that was the long-winded way of saying what they all said.
Awesome. Thank you so much. Really excited, like I said, about, kicking off this discussion. You all have, said dropped a lot of good nuggets for us to consume and consider for the discussion. So thank you again to each of you. So first, I want to probably ask an overarching question for all of you, because it sounds like there's some enthusiasm around master protocols. There's potential, but there's also a ton of considerations that may or may not make you a fan of them. So why would you say we need master protocols for real world evidence? Again, thinking about acceptable levels of wanted versus unwanted variation, the various types of real world data sources that are available or not available to research sponsors or researchers generally. Why do we need master protocols to support real world evidence studies then? And
I'll jump in and just say, first of all, we need protocols for any real world evidence study. It's not just master protocols. So as Josh was saying, and as the FDA has shown, sometimes observational studies can provide substantial evidence of treatment effectiveness or treatment safety. And those studies should have a protocol which is pre-registered. And if you vary from your protocol, you should say why you varied from that protocol and be very transparent about why you chose the data that you why it was fit for purpose, how you didn't cherry pick, etc. So we need protocols in general. For the master protocol issue, it's about multi-center prospective studies. And all multi-center prospective studies need a protocol. But it's even more important when you're using real-world evidence as a major source of the data, because there is so much variability in not only how the data is collected and curated, but how the data is, how patients are treated at different sites. And so that kind of unwanted variation or noise makes the protocol less efficient in being able to detect a treatment effect, which is what the goal of it is in the first place. But I think, as Josh said, the first question is, do you need a prospective randomized study? Sometimes you don't. And I think increasingly in the future, we will not necessarily have to rely on prospective randomized studies once we get an infrastructure in place that allows us to have confidence that we're getting complete, accurate, reliable information that can be turned rapidly into real-world evidence.
Thank you, Mark. Anyone want to also chime in?
Yeah, I would agree. It's about the, really where it's needed is the multi-site studies. So if you're doing a distributed data study or a study, even a study where data is collected at multiple different sites, you want to make sure that it's collected the same way, because when you put it together, you're not going to have an interpretable result. But I mean, I would argue that we need harmonization on a broader scale, that having sort of common definitions for cohorts and for outcome measures will be beneficial, because a lot of times that the studies themselves for FDA approval aren't doing comparative effectiveness, but patients and clinicians are looking for that kind of comparative effectiveness information, and it's impossible to synthesize across studies. And I would agree with what Mark said, that you need protocols in general that are specified enough so that you can tell when there's differences between studies. But even better would be if we had some sort of common definitions that we're all working off of.
I'll just chime in and say, yes, I agree with all of this. And it reminds me of a thought I had, Jeff, while you were talking. And it's the quote that's often attributed to Mike Tyson, which is that everybody has a plan until they get punched in the face. Trying to deploy a multi-site study, I am told by my colleagues, can feel a little bit like getting punched in the face, because you run into these very real sort of friction points that you maybe didn't anticipate or couldn't have anticipated. And I think that's where something like, that's where something like a master protocol can help you a little bit. It gives you something to sort of fall back on and build from, particularly as you get into these situations where, as you were saying, Mark and Elise, you get these additional sources of variability that can make it tough to know whether a thing happened or not.
Yeah, understand. I think the protocols alone, right, are incredibly important for anything that we do that is intended for decision making, regulatory decision making, has to be protocol based. You've got to write it, you got to write an analysis plan, and you got to post it. I mean, I think we as a community have to just kind of get used to that's the way it works. If you're doing something internally and doing something fast, you do whatever you'd like. But if you want someone else to making decisions based on it, it should be protocol based. Now that's really different than writing a master protocol, hoping someone's going to read that protocol and be able to implement it somewhere else and has thought about how to implement it. It should be detailed enough that you could implement probably in the same type of data source. Implementing a different data source, someone's going to have to make some decisions there. When we were working within COVID, and there's a lot of good examples in COVID, and with Reagan-Udall, we were trying to build a protocol, and this was actually Sentinel's COVID protocol, trying to build it so that many different people could implement it in many different systems. That's a really different, that's a different use case. And I don't know how many, I would love to think through how many times we need that, where we want the community to all kind of take it and go, versus answering a question. Let's always agree we should start. I think we should start with a question. So for the question and then go from there. And I'll say, Mark, you triggered a thought. I mean, I think the US has an incredibly robust real-world data evidence ecosystem. Sentinel, I mean, look, I used to be with Sentinel and I was there from the beginning. It's an incredibly robust system. PCORNet is a pretty robust system. So it's not that we're lacking thoughtful folks who put a lot of time and an awful lot of money into these systems. We have to think about how to better use them. And there's NIH examples of these systems. So I think we're rich in opportunity and struggling with almost what to do with it, because we could, I don't know. There's the old adage, like, enough rope. Maybe I won't go all the way to that adage, but you can hurt yourself if you have too much leeway and too much data, right? Think of it that way.
Well, the US system may be wonderful, but it's still completely inadequate. So it is, PCORNet is not representative of the US population, and it doesn't pretend to be. If we're going to really tap into a much broader population like Sentinel does, although that even that is limited by which organizations are participating, We need to invest in an infrastructure that can be reused for multiple different kinds of studies so that we can get the value of that investment, you know. We had in a different Duke-Margolis meeting that talked about distributed clinical trials. The point was made that building this infrastructure the first time is neither cheap nor easy. And the value you get from these infrastructures come through reuse, but many of the infrastructures are never reused. And so we never get that benefit of efficiency over time. And I think that the other thing is about linkage. The amount of data that's being linked so far today is relatively limited. But, you know, we need to link our clinical data from the EHR with our claims data, with our laboratory data, with our genomic data or whatever omic data, and whatever other data that we can in order to get a really robust view of what the patient is and what's happening in the disease. And we have a long way to go.
Yeah, thank you. You know, a lot of what you all are saying and what I've heard from some of our opening remarks by our FDA colleagues, I think touches on my next question. So good science establishes procedures before selecting data sources, you know, thinking about the robust and abundant amount of data sources that one might select, which, you know, can contribute to good science or biased outcomes or what have you. But if the development of a common or a master protocol is dependent on the research context, how early do you select good, relevant, reliable, worthwhile data sources for a specific protocol? You know, thinking about real-world data sources that exist out there. How early or how important is it, or at what point is it in the study team's best interest to select worthwhile data sources, you know, specific to building a master protocol?
Well,
I mean, I would think
It depends on the question. You know, it's what the question, you know, you have a question, then you figure out what your protocol should be, the ideal protocol you'd like to do. And then you see what data sources are available and if they can support that protocol and analytic plan. Mm-hmm. So, you know, it's in order. It's not, you shouldn't say, I have access to these data sources, now I'm going to write a protocol. If your goal is to develop generalizable data about, say, the US population, you want to be able to make it generalizable to the US population, then you need to have data sources which sample a randomized portion or a representative sample of the US population. And then if you do that, you can talk about that. But if you don't do that, and you say, oh, well, I can only do a multi-center trial and I got this group of investigators, which I've used before, then you're just doing another standard RCT with a lot more noise built in.
Yeah, at least you were going to also add a comment.
Oh, well, yeah, I mean, I would think you would definitely need to write the protocol first and say, this is what I need, and then look for a data source that matches that. But of course, if you, you know, once you start looking at data sources, you might have to say, well, maybe I have to make some compromises. But also, just going back to the point also that Mark made about linking, and I saw in the Q&A, there's also a comment about having a universal patient identifier that really, I mean, there's a big problem in the United States, which is that patients themselves can't aggregate all their own data with ease, and that physicians don't have access to all of a patient's data. So ideally, when you go into any physician's office, they should have information about all the other physicians that you saw, all of your tests and results, you know, that all of that should be at hand. And if that was true, then that data could then be de-identified together in a package to create a research data set. So, I think that again, I would just really stress we have to work together with other people who are trying to solve this problem more broadly.
Absolutely. Well, thanks. Okay, well, we will go ahead. We've got about 10 minutes left before we move into the break for our event. I want to remind members of our audience to submit any questions you have for our panelists using the Zoom Q&A function. That being said, I'll move into my next question. So I presented a slide earlier describing where master real-world evidence study protocols will have perhaps the greatest utility, and that's particularly in considering pragmatic trials or point-of-care trials, where we're aiming for leveraging master protocols to achieve high study precision and accuracy and high study efficiency. So I wanted to talk a little bit more about that. What is your perceived relationship between real-world evidence master protocols and executing or building pragmatic trials, especially those that contain point-of-care trial sort of operational components?
I guess I'll start us off. I think it makes a lot of sense to talk about those together, because when you think about doing a pragmatic trial, or when you think about sort of the desired future state that grows out of something like that, and this is something that the Clinical Trial Transformation Initiative has worked on, the idea of integrated clinical research into clinical care, where it's sort of the seamless coming together, and as Mark called it, a learning healthcare system. When you think about generating real world evidence, or generating evidence that can inform decisions based on the stuff that you're already doing anyway, I think that master protocol becomes really important for a lot of the reasons that we've talked about, managing not only the variability in how data are collected and recorded, but even in standard of care, that very slippery term, where what standard of care in my hospital, if I'm a practicing clinician or clinician investigator, may be different than the standard of care, say, at an otherwise very comparable setting for a colleague in the United Kingdom, to pick an example. And we saw some of the issues regarding comparisons brought to of that type, brought right to the fore as we all tried to make decisions and reach some knowledge together about what to do about SARS-CoV-2. So it was a great opportunity to highlight how useful it is to say, if we're going to try to do studies in that flow of providing care in a pragmatic trial, for example, or a point of care trial, I think my feeling, or my opinion, is that it becomes all the more important to make sure that we're all that everybody that's participating in that combined research and care providing effort is going to be doing enough of the same things that at the end of it you are able to draw a robust conclusion, and really beyond that, compare it to other robust conclusions that have been drawn from similar studies. So the TLDR summary is that, I think it's really important to build some of that master protocol concept into pragmatic and point of care trials.
I would jump on that and say it's not only important, it is absolutely critical, because a pragmatic and a point of care trial turns very rapidly into a platform trial or a basket trial. When you're in a standard RCT, your randomization is enforced. You can't change your treatment, and you're monitored to make sure you're taking the treatment. And so that gets us, that internal validity about whether we're seeing a treatment effect. In a pragmatic trial, on day 2, you're ending up with what's more or less an observational study. Because if it's not working out, the physician's going to say, this is not working out. I'm going to change the dose, or I'm going to change the medication, or I'm going to add something. and suddenly you now have a different subgroup. And the other thing is you have a much wider range of patients, whereas in a clinical trial, we're only taking stage 2 or stage 3 patients. In a pragmatic patient, you're taking everyone from stage 4 to stage 17. And so that alone is different trials right there. So in essence, you've created a platform or a basket trial, and you need to think in your protocol about how you're going to deal with that variability as you do your analysis before you do the trial, not after you start collecting data.
Yeah, thank you. Well, we do have some questions from our audience members. I'll start with one question that we got that I think is a really good segue to our discussion. One of our audience members asks, in cases where there is no common data model in place, how could a master protocol in practice be applied? It seems a bit wishful, seems a bit of a wishful thought, and a very questionable relevance. Would anyone like to respond?
Describing the old days. That's the pre-CDM world. So I'm going to go back to the same example. If it's a protocol, to completely unrelated people and two completely unrelated institutions never speak to each other and try to implement that protocol independently, they won't do it the same way. And how much variation they'll do, it's hard to know. It depends on how specific the protocol is. If they talk to each other, you have a much better chance of it working. But the reason we built all those common data models and all those networks back to the HMO research network and the vaccine safety data lake and all of those was because 10 programmers coding against the same protocol did not code it the same way. Full stop. It just never happened. So that's why it's the and we've done even, Centone has actually done some nice research showing where you would see variation if you have two thoughtful folks in the same document. They published some of that. So unrelated to each other is really hard. Related to each other and speaking, much more likely to generate something that's comparable. I've been through the pain of that. Maybe you can hear that in my voice.
Yes. We heard it earlier too.
Yeah, tried.
But I would also, just sort of like push back a little and say, okay, but different health systems aren't doing the coding the same way. So even if you have like a code list and you say, this is my definition of asthma, it still might not be pulling the same people at the same, at different systems. So so I think that this is where more research is needed. But if you did have a local definition, you say you give the clinical definition and you have each site pick out the code list that matches the coding practice at their site. Is that more accurate? Or then even the future, when we go to AI machine learning, if we did implemented AI machine learning at different sites to pick out cohorts, are those more similar or less similar than just having a standard code list?
So you're absolutely right, Elise. Even if they're using the same definitions, different healthcare systems do not use the same mechanism for transforming their data and curating the data. And so there could be subtle variations in how they transform, curate, what they do with missing variables, what they do with null variables, et cetera, that lead to differences. So the common data model at least tells you that they've looked at what the data that they produced, and they believe it means X. And so the X of what it means is what somebody can aim for. In other industries, they don't necessarily use common data models. They use translation layers because they're just making sure it's translated into the X of what they think it means. But the common data model is important because we have such arcane terminology in medicine that it makes sense to have a common data model.
No, we're right at time, Rachel. We're sounding a little depressing, but you know, we're describing all the stuff that are reasons it's hard, but it's really important to say it's not so hard that we can't do it. We know enough to do this well. We just have to be able to do it thoughtfully. There's no definition of diabetes. It doesn't exist, period. There's 100 definitions of diabetes. You just have to pick the one that matches your research question and tell people what you did. But there's 50 years of experience using real world data. This year, it's 50 years of the first paper. So, you know, we know why it's really hard, which is why you asked us to speak, but we have no choice but to use the data we have and not the data we wish we had, and the systems we have and not the systems we wish we had. And I think we actually have really pretty good systems. So I don't want to let us go. Sorry to take you over a minute, but
No worries.
I was feeling, you know, down. We're not so bad.
No
As a
worries.
community.
Well, that's what.
Very well said, Jeff.
Agreed. Yeah, that's why we're here to have this discussion. You know, we're not necessarily championing one effort over the other, but we're opening this up for discussion to, again, get everyone thinking about these issues and talking about them. And so we're glad that you all were able to do that today. And we certainly look forward to more discussions on this, especially with the dissemination of our Real World Evidence Master Protocols white paper. Again, I encourage everyone here to take a look at the white paper, the advanced copy that was shared in our webinar chat through a hyperlink. At this point, we are going to take a break. We will still be live during the break time. So, you know, as a reminder to our panelists, and, you know, please, you know, feel free to go off camera and stay muted, and everyone in attendance, please join us back here at 2:25 p.m. Eastern as we continue with the day's discussions. Hi, everyone. Welcome back from our break. We're glad that many of you are able to continue joining us for today's event. We hope that so far, everything that we've shared thus far has been informational and of interest to everyone here. We're glad to still have quite a sizable audience with us for the remainder of the day. So with that being said, we are going to go ahead and move into our session 2 panel, which is focused on evaluating real-world efficacy in patient subgroups. This session will address opportunities and challenges to leveraging real-world data in order to estimate and measure treatment effects among patient subgroups, with a specific focus on terminology considerations that accompany measurements of therapeutic effect in subgroups, and leveraging real-world data to address data missingness about patient subgroups and improve care more generally among them. So before we move to our panelists who will provide opening remarks, as did our panelists in session 1, I'm going to turn our presentation over to Nora Emmett on our team, who is a senior policy analyst. Nora will provide a brief overview of our recently released and finalized white paper entitled Improving Patient Subgroup Representation with Real-World Data, based on takeaways from our December 2022 convening and workstream effort focused on real-world efficacy in patient subgroups. So bear with us for one moment as we prepare our slides for Nora's presentation, and I will go ahead and turn it over to Nora.
Hi, everyone. As Rachel said, my name is Nora Emmett, and I'm the Senior Policy Analyst here with the Real World Evidence Collaborative. And before we have our panel discussion, I'll give an overview on our work with patient subgroups in real-world efficacy. And we can go to the next slide. Thank you. Today, I'm going to go over some of the policies related to patient subgroups in real-world evidence that led to the creation of our workstream. I'll go over the workstream and the white paper we developed that was published today. And we can go to the next slide. And the next slide. Okay, so I'll give an overview on FDA regulatory policy relating to patient subgroups. In 2014, they put out an action plan to enhance the collection and availability of demographic subgroup data with 3 priorities to improve the quality, participation, and transparency of subgroup data. There was the April 2022 draft guidance on diversity plans to improve the enrollment of participants from underrepresented racial and ethnic populations in clinical trials, where FDA called for all new drug applications to present effectiveness and safety data stratified by gender, age, and race. And just this August, they put out the draft guidance on post-marketing approaches to obtain data on populations underrepresented in clinical trials for drugs and biological products, where they mentioned that disease occurrence and outcome can vary based on demographic factors. Okay, we can go to the next slide. And I'll quickly go over relevant information from Cures 2.0, specifically Section 203 on increasing diversity in clinical trials. The Act calls for increased study into barriers faced by underrepresented groups to clinical trial participation, public awareness campaigns aimed at minority communities designed to increase knowledge of upcoming and ongoing trials, and the creation of a permanent task force to make clinicaltrials.gov more user-friendly. And we can go to the next slide. And the Prevent Pandemics Act called for an improvement in the collection, availability, and sharing of public health data, with specific mentions of increasing data quality and capturing demographic information in culturally and linguistically appropriate manners. We can go to the next slide. So I'll go over our 2022 work stream on real-world efficacy and patient subgroups. And we can go to the next slide. So as I mentioned in the policy overview, regulators like the FDA are prioritizing the collection of subgroup data. There's really great value in collecting this data, as underrepresentation of subgroups with specific demographic and clinical characteristics can limit the reliability and validity of data, as well as exclude these groups from all benefits derived from research. And certain subgroups are traditionally excluded or otherwise unengaged in RCTs and other forms of clinical research. And to address these issues, our workstream explored opportunities and challenges to leveraging real-world data to assess treatment efficacy. And we can go to the next slide. So I'll now share an overview of the white paper. We can go to the next slide. The white paper is titled Improving Patient Subgroup Representation with Real-World Data, Real-World Efficacy in Patient Subgroups. And this white paper was informed by our workstream discussions and a private workshop held last December focused on terminology considerations, methodological challenges, opportunities to use RWD, improving race and ethnic subgroup data collection, and point-of-care trials and patient advocacy organizations. Okay, we can go to the next slide. The white paper included the sections here on the screen, and I'll give more detail on a selection of these in the following slides, with a specific focus on terminology considerations, using real-world data to supplement clinical trials for subgroup analyses, leveraging real-world data to address shortcomings in trial data concerning racial and ethnic subgroups, and leveraging real-world data to improve care amongst patient subgroups. And we can go to the next slide. To begin, there are a number of terms important to real-world efficacy. They're often used interchangeably. These terms include efficacy and effectiveness, and subpopulation and subgroup, which are all distinct concepts but are interrelated. Alignment on these specific terms as a necessary first step to enable shared use, prevent potential misunderstandings, and promote collaboration among providers and researchers in practice. And on the slide here, you can see practical considerations and policy recommendations for each set of interrelated terms. Give me a moment. Okay, we can go to the next slide. When using real-world data to supplement clinical trials with subgroup analyses, there are methodological challenges necessary to consider, and these include the lack of structured and unstructured data standardization, variation in data quality and accuracy, variability in demographic stratification methods, challenges with accessing real-world data sources, and the limited acceptance of real-world sources within regulatory contexts. Let me go to the next slide. And there are several best methodological practices to take to address these challenges when leveraging RWD for subgroup analyses. A few I'll highlight are utilizing high-quality real-world data and evidence sources appropriate study designs, and statistical methods, as well as using study designs with pre-specified protocols and clearly defined endpoints to in turn reduce the likelihood of inflated results and type 1 statistical errors, and validation, both internal and external, of real-world efficacy and outcomes in post-hoc studies using RWD. Okay, and we can go to the next slide. Another strategy to using real-world data to supplement clinical trials is the use of ex-US data. Ex-US RWD is RWD collected outside of the US, and this was of particular interest during the emergence of COVID-19, and trial sponsors since then have used RWD to improve ex-US RWD, to improve access to patient populations that are challenging to recruit here in the US, to access samples of patients unfamiliar to specific elements or treatments within the US healthcare system, to navigate limitations and challenges in RCTs, and to support updated labeling guidelines. And it's really important to note here that the value and utility of ex-US RWD should be interpreted case by case and should not be used as a strategy to circumvent any trial outreach or engagement with underrepresented subgroups. And we can go to the next slide. So race, ethnicity, ancestry, and genomic data are important in healthcare research, as the interaction amongst person's biology, environment, and social determinants can affect their overall health, disease status, and risk, as well as any responses to drugs or medical products. And oftentimes in healthcare contexts, these data are missing or not considered, even at times where it could be argued that this data is necessary and actionable, particularly for high-risk subgroups. Leveraging RWD can be a potential solution to address these data shortcomings, especially amongst racial and ethnic subgroups. And researchers and trial sponsors can turn to RWD or E generated during phase 4 RCTs to address this missing data and determine whether real-world efficacy or treatment effects might differ between racial or ethnic subgroups that were either overrepresented, underrepresented, or unrepresented in RCT phases 2 and 3. And RWD/E can be a strategy to broaden inclusion criteria and limit unnecessary exclusion criteria, which will then lead to more diverse patient cohorts, to more accurately assess treatment effects amongst different subgroups, and a reduced likelihood of biases and observable treatment effects. Okay, we can go to the next slide. And certain patient demographics are overwhelmingly underrepresented in clinical research, which largely includes racial and ethnic minorities, sexual and gender minorities, and pregnant and lactating persons. And to address this subgroup data missingness, there are a number of best practices for leveraging RWD and E, including using categorical demographic terminology consistently and appropriately, supporting data quality with IT infrastructure, and communicating how and why data will be used, and sharing results back to communities. Okay, we can go to the next slide. Point of care trials are one way to leverage RWD to improve care amongst patient subgroups. Point of care trials are an operational approach to conducting pragmatic trials in healthcare settings and can be leveraged to recruit and engage diverse patient subgroups to help improve generalizability in trial results. And this work can help bridge evidence gaps, but more is needed to ensure that these trials are conducted routinely. And furthermore, trial sponsors must first address barriers to trial participation faced by underrepresented groups. And these researchers need to implement strategies to receive and include input from the communities they work with into their work to ensure return of value. Okay, we can go to the next slide. And I'll just briefly touch upon some key takeaways from the white paper prior to the panel discussion. Okay, so challenges to measuring real-world efficacy in patient subgroups are met with opportunities to leverage real-world data to observe and measure unique characteristics amongst patients, more closely assess treatment options available to them in the real world, and help clinicians make more informed decisions and provide more personalized care. And policies that are related to the research and development of treatments that prioritize equitable inclusion of subgroups which have previously been underrepresented in patient research, clinical research, sorry. Those are essential in order to ensure that medical advances are informed by all people who may need them. And finally, trial sponsors and other RWE stakeholders should create opportunities to learn from the latest developments concerning personalized medicine. They should engage with local healthcare providers and community leaders located in areas where these underrepresented clinical subpopulations live, and establish sustainable recruitment sites and partnerships in those areas. And with that, I will hand it over to Rachel to just introduce the discussion panel. Thanks all.
Thank you so much, Nora. What a great overview. It's my pleasure to move into our panel discussion portion of this session. I would like to ask our panelists to please come on camera and unmute yourselves as we begin. Thank you, and we're glad to have you all with us. It's my pleasure to introduce each of you. Our first panelist is Evelyn Hudson. She's the Global Real-World Evidence Director at Novartis Global Business Solutions. Our next panelist joining us is Greg Caleb, who's the Director of Global Epidemiology and Oncology at AbbVie, and also Adjunct Associate Professor at the USC Alfred E. Mann School of Pharmacy and Pharmaceutical Sciences, their program on medicines and public health. Our third panelist joining us is Daniela Moga. She's an Assistant Dean for Research and Larry H. Spears Endowed Chair in Pharmacogenetics in the College of Pharmacy at the University of Kentucky. So we're glad to have each of you here with us. And I will go ahead and open up our panel with a few opening remarks from each of you. So first, I'll turn it to Evelyn, who will provide her opening remarks first. Go right ahead.
Thank you so much, Rochelle, and thank you to Nora for such a lovely summary of the white paper, and thank you to all for having such a collaborative. So I suppose the first point that jumps to mind is with regard to the FDA. The FDA's call to have more diversity and inclusion in clinical trials, and I think this is something we can all get behind. And obviously, I'm a big fan of real-world data, but would really like to also see the more subgroups being represented in clinical trials as well. It's been mentioned a couple of times today about having fit-for-purpose data, and this is also very true when it comes to subgroup analysis. So having data in which you can accurately identify the subgroups that you're interested in examining is really important. The other point, and again, it's probably been echoed in other people's presentations, is about that pre-planning. Real-world data is a lot like a box of chocolates. You don't know what you're going to get. And sometimes, well, most time it helps to do a lot more thinking and planning upfront and thinking about that, the results you might get, the analysis you can get, so that you can have a really robust protocol upfront before you do the data. And if you are in the fortunate position of collecting data, to think about how you can better represent different types of subgroups, and think about the different granularities that you might want to look at in that data. I mostly work with secondary data, and sometimes I wish, I wish I had data on X. And where possible, to leverage internationally validated definitions of subgroups, and to continue our efforts to try and identify these different definitions. Sometimes with something as simple as maybe choose the example of smoking, where you think that would be a fairly standard variable. I've often seen data where you could have patients in year one are smokers, and then in year two they're down as never having smoked. So how do you code that up in your data? So to try and think of these different different possibilities that you might see in your data. So trying to collate these different definitions. I think it was mentioned in the previous panel that you could have different definitions for different diseases, but to try and identify, well, okay, I might use definition 1 for this circumstance and definition 2 for this circumstance, but to have that available as a resource for researchers going forward, I think will be enormously beneficial. And I suppose my final remark is, I mean, at the end of the day, I suppose what I certainly think about, and I'm sure other people on this call are thinking about, at the end of the day, you're wondering, is this treatment or medical device, is it going to work for people like me or people like my patients? And that, I think that's the question we all want to know. And this collaborative hopefully will help move that in the right direction. Thank you for allowing me to make some comments.
Thank you so much. That being said, we will go ahead and move to our next discussant. Greg, feel free to take it from here.
Great. Thanks very much for the opportunity to join all of you on this panel. It's really an honor. So I'm going to be discussing real-world efficacy in patient subgroups in the context of racial and ethnic inequities and the external validity of clinical trials. Much of my own research is in real-world evidence on oncology, so I'll be referring to a few examples in cancer clinical trials. Next slide. So as we heard in the report on the white paper, there's currently this heightened awareness of underrepresented populations in clinical trials. And part of that is attempting to understand why that is. So evidence in patient subgroups may reveal important factors related to efficacy and safety of cancer treatments, and underrepresentation and lower participation of patients of color directly constrains that ability to look at patient subgroups. It also undermines the external validity of the evidence driving our clinical and regulatory decision-making. Where that's relevant for us here today is that part of the promise of real-world data and real-world evidence is that it has an ability to fill those gaps. So the study that I'm showing here below is a real-world evidence study on racial and ethnic inequities in U.S. oncology clinical trial participation from 2017 to 2022 that used the nationwide Flatiron Health database. And basically it showed that in the last 5 years, oncology clinical trial participation was lower among Black and Latinx patients relative to white patients in this network. And there were apparent differences where there was even widening of that disparity in the years of the COVID-19 pandemic. As Nora described earlier, in 2022, the US FDA disseminated draft guidance on improving enrollment of participants from historically excluded racial and ethnic groups. In addition to driving sponsors to develop really intentional and equity-centered diversity plans, it also represents an opportunity for real-world evidence to help inform the design of trials and also remain this core component of post-marketing requirements and commitments that, again, fill these evidence gaps. Next slide. The data here were presented at this year's ASCO annual meeting and the annual meeting of the International Society of Pharmacoepidemiology. This was actually an extension of that prior study from Flatiron Health. And I'll just let you know the main takeaways here were that, again, Black and Latinx patients were underrepresented, about 40 to 50% less likely to participate in cancer clinical trials, but also that social determinants of health explained about 50 to 60% of the racial and ethnic trial inequities that were observed. So these included factors like insurance status, where patients were treated, and at the area level, whether they lived in areas with racialized segregation, low vehicle ownership, and limited English proficiency. So with respect to what this real-world evidence offers to the development of diversity plans, we understand that addressing these supportive services that mitigate language and transportation barriers, these are important. Understanding where our clinical trial sites occur, whether they occur in communities of color or not, that's also important. There's also similarly complex barriers to health equity in evaluating real-world efficacy in patient subgroups. So, for example, in multiple myeloma, there are inequities in access to cytogenetic testing, and this represents a barrier for both in clinical trials, a path to be eligible and participate in those clinical trials that are biomarker-driven. It's also pervasive into our real-world evidence, and then it makes us less able to evaluate the effectiveness of therapies in real-world data when these patients lack this kind of documented testing altogether. So kind of a core at where we are getting our real-world data. Next slide. Another example I'll just touch on briefly comes from a recently published article in Blood by Casey and colleagues at Medical College of Georgia. It really highlights where this underrepresentation really kind of goes awry. The authors describe underrepresentation of historically marginalized or minoritized groups and by geography in lymphoma clinical trials. and those that resulted in a regulatory approval in the last 10 years. So for example, here in non-Hodgkin lymphoma trials, the enrolled populations were only about 2.8% Black and 5.4% Latinx. The target population of lymphoma, or the burden in the US, is Black patients making up about 10.7% of lymphoma patients and Latinx patients making up 17%. So in the context of real-world evidence studies, barriers also exist to inclusion of patients of color. For example, studies that use administrative insurance claims data, we often apply requirements to continuous enrollment on health plans, and we know that historically marginalized or minoritized groups are typically have a higher rate of being uninsured or underinsured. So taken together, I'm looking forward to discussing today how we can really fulfill that promise of real-world evidence in representing all patients and really addressing racial and ethnic inequities that we know are pervasive.
Excellent. Thank you so much, Greg. I will turn it to our final discussant, Daniela. Feel free to take it from here.
Thank you so much, Rachel, and thank you, Duke-Margolis, for the invitation. I would also like to thank my fellow presenters for setting the stage and making a compelling case for the importance of considering patient subgroups and evaluate treatment effect heterogeneity for different patient subgroups. Next slide, please. So current recommendations for assuring diversity in clinical trials and studies using real-world data are based on the premise that people may experience the same disease differently or respond to the same treatment differently. It is therefore essential to include people with a variety of lived experiences and living conditions, as well as characteristics like what we define as race and ethnicity, genetic profile, age, sex and sexual orientation, gender identity, pregnancy status or comorbidities, so that all communities can benefit from scientific advances. I will take the next few minutes to share a few thoughts that build on the previous presentations, reflect on my own research experience addressing heterogeneity of treatment effects, and highlight the importance of developing standard procedures for data collection to allow for accurate evaluations of heterogeneity in treatment effectiveness, as well as the need to consider components of diversity that might not be fully captured in the existing paradigm on how we think about patient subgroups. First, I will discuss some issues surrounding the way biological sex, also referred to as sex assigned at birth, versus gender identity are captured or maybe not captured in real-world data like administrative claims. And I will follow with bringing to your attention cognitive health or brain health as a lesser known factor that can impact effectiveness and should be considered in defining patient subgroups in some situations. Next slide, please. In an effort to evaluate health outcomes in non-cisgender adults, we use the comprehensive approach based on multiple types of data, including inpatient and outpatient claims with diagnosis and procedure codes, pharmacy claims, and enrollment information captured under the variable sex. Specifically, the algorithm, which you can see referenced on the bottom of the slide, uses this data to identify codes for identity disorder or codes that are indicative or gender affirming therapy. Further classification within the broader category of non-cisgender people require comparison against the variables suggestive of sex assigned at birth. But what we found when applying the algorithm to US employer-based insurance claims data was an increasing trend of non-cisgender adults that are being included in what is classified as unclassified category. While in data analysis, we usually exclude information we don't know anything about. You can see that this is quite a big proportion of the data. And we could then go and just analyze using these three categories. But I would argue that we need to consider the heterogeneity that might be inherent to this unclassified group. Some of the people who fall into this category might be transgender women. They can be transgender men, non-binary people, or transgender individuals with another gender identity who have not accessed medical forms of gender affirming or do not plan to do so. And this is relevant when thinking about evaluating disparities in care or treatment effectiveness for conditions that disproportionately affect people identifying as non-cisgender. And we know that information captured under the variable sex in insurance enrollment files might be updated at intervals that correspond to a change in insurance status. It is unclear whether data collected from different employers that feed into the insurance enrollment files reflect one's gender identity or sex assigned at birth, thus making it difficult to accurately identify individuals from these large data sources, which would be important and would aid in generating valid real-world effectiveness in non-cisgender subgroups. Next slide, please. We will now move into discussing cognitive health and the heterogeneity inherent to this. Clinical trials typically aim to select homogeneous groups to measure efficacy. However, it is more subtle characteristics like the rates of cognitive decline that might not be fully captured. And you cannot just assume that randomization will achieve perfect balance. The potential for selection bias would therefore affect study design. And furthermore, this heterogeneity, if it's not considered, then treatment would be assigned based on a heterogeneous group and would therefore impact the outcomes when the treatment is used in real world. Next slide, please. I will use another example on the importance of considering heterogeneity with regard to cognitive health. And this is in the design stages of real world studies. The example I'm presenting here is not necessarily related to a specific medication, but rather to an intervention that is considered a treatment optimization approach or a deep prescribing intervention. I believe this example is quite relevant for the point I'm trying to make. Here in this study, we evaluated the cognitive impact of a deep prescribing intervention. We measured, aside from cognitive function, we measured medication appropriateness, but I will only focus on cognition. And this was done by enrolling a group of participants without symptoms of cognitive impairment that were randomized to the intervention or standard of care. We hypothesized that our intervention would bolster cognitive function with a bigger effect in those in a preclinical Alzheimer's disease state, as was identified based on a brain scan at baseline identifying amyloid deposits. What you see on the top of the slide here is the overall effect when we did not take into account heterogeneity of that preclinical Alzheimer's disease state. And there was no effect to be found on the intervention and its impact on cognitive function. If you look at, if you can click one more time, I was going to the next slide. So this is the point I was trying to make with seeing no effect on cognitive function if we look at the group overall. Next slide, please. But now, if we are looking at, and you can click one more time. Thank you. If you look at this bottom part of the slide, you can see that when we took into account the stratification based on preclinical Alzheimer's disease status, we can see that there was an important impact of the intervention on cognitive health after the intervention. These are the examples I wanted to share just to kind of build on what was already said and bring some other points maybe for your considerations. I hope this helped emphasize not only the importance of considering patient subgroups in the evaluation of real-world evidence, but also the need for strong methodology and standardization in our work. And with that, I thank you for your attention, and I will hand it back to Rachel and Nora.
Wonderful. Thank you so much. I think I anticipate this being a very robust discussion, as this topic typically is most times. So I'm happy to go ahead and move into our discussion portion of this session. And I want to remind everyone here in attendance and our audience to please feel free, again, to use the Zoom Q&A function or feature to ask our panelists questions as we continue with the discussion, or as any come to mind based on what you've heard so far from our panelists. So thank you again, Evelyn, Greg, and Daniela. All of your opening remarks were incredibly informative. Thank you to Nora for your overview of our white paper. Again, if anyone would like to take a look at the final release of our patient subgroups white paper, it is available in the webinar chat. We have a hyperlink to a full PDF, and feel free to follow along as we continue this panel discussion. So to kick off this session discussion, I have a few questions for you all, but I want to start by first asking a really important question that comes up a lot, or that has come up a lot even among members of our Real World Evidence Collaborative this year, and also within the broader Real World Evidence policy stakeholder community, who's really, as many have mentioned already, really concerned about data quality or cleanliness, or our ability to appropriately clean data when we find discrepancies in one source versus another about a particular high-risk subgroup. And so my first question is, what are your thoughts as far as how researchers can address data quality in work that they undertake with assessing efficacy, drug or treatment efficacy in the real world regarding a specific subgroup, whether or not they are high risk, or there's high penetrance of a disease based on a biological factor or characteristic, especially subgroups that are already underrepresented in clinical research. What are some data quality considerations that should be addressed?
Well, the first thing that kind of comes to mind when you're saying there with data cleaning, etc., is, I suppose, that the provenance of the data. So that we have a clear record of how you got, any steps that were taken in terms of when the data comes your way, depending on what stage of the data collection process you're at, to the results that are produced. So if a record is imputed, if something is deleted because it appears to be noise or whatever, having a clear record of anything you do to that data, so that if ever you need to come back to it, that you can be assured of so that's one aspect of quality that kind of comes to mind for me with respect to the data, so that people can trust that the data they enter in is what they get. So that's, I suppose, the first thought that comes to my mind with regard to quality. I'll also, I suppose, harp on again about standardization, and if there are defined definitions of whatever subgroup you happen to be looking at, and to be upfront if there's any reason why, for whatever reason, respect to your research question, that you're deviating from that definition, or if in fact you need to create a brand new definition, what was the rationale for how you categorize the different subgroups, etc. So I think transparency is really key.
Wonderful, thank you.
I would like to add to what Evelyn said, and I fully agree with everything that she mentioned. It's kind of like made me think maybe back to the keynote we had earlier today, where Dr. Rivera really showed that nice image of like the data, what we see, right? And kind of going back to who's behind that data, right? And so kind of understanding what exactly is being collected. And I think I'm hoping that, my example with, is it sex assigned at birth, or is it, gender identity? We see more of maybe in some situations less stigma, so people are more open into being, reporting on their gender, especially if they underwent gender affirming treatments. And so we don't know exactly what is being captured in those data. And it's so basic. Like when I started in this area, I always assume that what you find in enrollment files is sex assigned at birth. Whereas we see now that it might not be the case. And the fact that we aren't clear and there is no standardization on how these data are collected is an important consideration for the future, and maybe a call for more work and more effort into understanding those data that are not necessarily collected for research, on how are they being collected and what is exactly available to researchers.
I think that's a really great comment on, what becomes available. I think taking sort of a health equity lens to this question, and I guess I'll put it in the context of oncology real world data. So in this space, in cancer real world data, it tends to be highly curated real world evidence, those that require abstraction and other types of technology, machine learning, and natural language processing. There are core parts of that are also, where health equity gaps can become a real problem. And so I mentioned before that of, if you're using insurance claims, a condition there is that patients have insurance and that they continuously stay on insurance and then can be observed in an observational study. There's quite a few others. The same with respect to equity in insurance status and genetic testing. So, we often don't think of those things in terms of the completeness of data and, equitable or quality real world evidence subgroups. Some of those solves or solutions are maybe related to kind of insurance and robust health insurance and, equity in genetic testing, things like that. And so those are also part and parcel to this discussion of real world evidence.
Thank you all so much. We do have a question from an audience member. Are there any unique considerations for subgroups with real world data or evidence generation and its use specifically as it relates to over-the-counter products?
I will give it a try to begin with. I think over-the-counter are quite tricky, right, to study, and the fact that we don't rely as much, when we don't have as much data that we can rely on, right? You can go and buy it, nobody knows you did. And so really hearing from patients, and I don't know, kind of maybe expanding on the information that is being collected as part of the healthcare to understand what patients are taking outside of the prescription medication. So maybe EHR could capture a more of the OTC use. I know there are barriers to even doing so with regard to time and focus of the patient visiting their physician or other healthcare professional. So I think there are definitely different considerations for the study of over-the-counter drugs. And the first thing that comes to mind is really availability of information on the use of those medications and the impact on health outcomes, and how different subgroups would respond to those is an important thing.
Yeah, and I think I'd like to ask probably more along the lines of that question, thinking about underrepresented patient subgroups and whether or not they might, provide over-the-counter medication information to their healthcare providers such that it is therefore documented within the EHR. Thinking about other, the wide range of real-world data sources to consider to capture that type of information, especially about or concerning subgroups that might have a low number of encounters or that otherwise might have less healthcare utilization, perhaps for purposes of not being able to affordably or reasonably access healthcare. Are there other real-world data sources that come to mind based on your practice that might convey that level of information about underrepresented patient subgroups?
So I kind of took one relation to the original question, which is potentially recall bias and people with multiple morbidities, that if they're taking many things, that there could be kind of issues there in terms of representativeness. There's one data set that comes to mind. It happens to be an Irish data set where it's a representative data of people as opposed to patients, And one of the things they do there is to go out to people's homes and they ask them to show them the medications that they have in their homes. And they list that as part of the data. So that's a really unique, or unique to me anyway, my experience of data that may not be so commonly done in the wider research community.
Really interesting. And I think touching on kind of both with respect to kind of the availability of this information and maybe with underrepresented patients too, you know, when we think about over-the-counter products, and I guess as a former pharmacist and current epidemiologist, I think about this kind of immeasurable bias that comes from that. You know, I think that there are places in which we could do better to record it and probably make these more available too. So if, say, for patients with Medicaid coverage, if all of these over-the-counter products were covered by Medicaid, it would open that window for us to understand whether or not patients are using them and patterns of use and what importance that has on overall population health. I think that that's one possibility. Even the things like FSA dollars being able to be covered for that too. It's kind of these other windows into kind of our patients' health and, you know, the medication use. So I think that there's a lot of opportunity there. This is a great question on over-the-counter products.
Definitely agreed. Yeah, thank you again to all of you for answering that question. I want to talk a little bit about something that you just brought up, Evelyn, about, you know, some things being capturable in the Irish data set versus the an American data set. So given that different countries will categorize different patient subgroups differently, thinking about, you know, the fact that some countries, and also thinking about within the scope ex-US data as Nora presented earlier, you know, some countries don't collect specific demographic information related to race or ethnicity like the United States does. You know, what are your thoughts as far as reconciling that with US data sets and or supplementing US data sets in ways that would allow perhaps for more generalizability about underrepresented patient subgroups in clinical trials?
I suppose any efforts that are undertaken to have kind of standard international definitions of these different demographic factors will really help that in terms of those planning data collection efforts to have to think really broadly about how their data could be used. It will be really key. I'd also add that even sometimes when you have the exact same items and response categories, they still might be interpreted in different ways. So you still have that potential for reporting heterogeneity. I have experienced in the past where we've had an Irish database and a US database where we've tried to harmonize the questions that were asked, and you still end up with different interpretations of seemingly innocuous questions. So being cognizant of cultural sensitivities is really important as well. So I suppose involving as diverse a group in terms of informing how you go about forming your data collection process, I think might help with that as well.
Thank you.
I will also add that, you know, sometimes maybe we have to, you know, think back to the research question we are trying to answer and decide whether it is fair and you know, would really help answering the question of bringing data from various, whether the noise from the heterogeneity that's brought from these different data sources is complicating, right, the matter. So I think maybe we have to consider that and decide whether every single question needs to be answered by combining the data sources versus using more of a stratified approach that allows for a more homogeneous interpretation within those different you know, countries. Maybe questions that rely on heterogeneity with regard to more biological aspects of diversity and heterogeneity are easier to answer than things that are heavily influenced by cultural background and also sensitivities to answering a specific question. So I think I see all of this as not a one-size-fits-all approach, but really rather thinking through what exactly are the pluses and the minuses. How can we, bring up the strengths of each and combination versus creating more noise than it's necessary.
Good point. Greg, any thoughts?
Sure. I just want to add, and I think I would be remiss in not mentioning that, so race and ethnicity is, as a lot of folks in the US refer to it, is this very uniquely US thing, a social construct that is really kind of a reflection of structural racism and other kind of systemic injustices that have happened in the United States. With that said, those type of inequities do kind of exist across other spectrums. So, I can think about things in terms of measured socioeconomic status in the US and things that measure deprivation status in other countries. And maybe that's where some of the transportability of some of this information on kind of groups of interest, underrepresented groups, folks that are prone to some of those kind of systemic injustices, things like that, where we can identify if that is part of the research question of interest. That's often what we think of with respect to race.
Indeed. My next question, I think, is probably related to, or certainly related to what's come up already concerning pregnant and lactating subpopulations, as well as sexual and gender minorities. So I want to talk a little bit about privacy, especially as, a lot of events have transpired that target these specific subgroups or subpopulations of individuals, either through violence or legal enforcement around abortion care, things like that. So what are privacy concerns or considerations when leveraging real-world data or evidence about these particular subgroups or other smaller subgroups like rare disease patients as well, even distinguishing between, privacy concerns that one might have over the other or not? What concerns come to mind or have come up in your practice as far as characterizing treatment efficacy in these groups, or even identifying where they are, so to speak?
I suppose generally, for whoever is collecting the data, I suppose there needs to be a lot of trust there, depending on what the item in question is, that the data is going to be anonymous, etc. You also might have situations where people, if they put down a category, they might be, maybe for insurance, they might be treated differently, etc. So there might be a lot, so stigma, being treated differently. Also, if a question is going to lead to a whole load of other questions, and that can be off-putting for people. So yeah, I think trusting the system, and we're all very conscious of data leaks and things like that. So there's a really robust data capture system to protect your data. And then once it's captured and protected, that when you come to do your research, that it's going to be reported in a manner that wouldn't reveal your identity, etc.
I will add that, stigma that's associated, or the fear of negative consequences with reporting certain statuses is quite key. And I think for us, as I'm thinking about myself as a researcher, we often are so focused on just our issues of misclassification and selection bias and things like that. So I think thinking and stepping back and going back to who is behind the data we are analyzing, it really puts a different lens on the issues of privacy and how you treat those data. So I think we need to maybe do a little more education in general with this, and how to build on the trust, maybe that we are really treating this for the benefit of the group, and we need this information, because to kind of close the disparity gap, it's important to be able to really look into the problems that affect that subgroup and find solutions that will be aligned. And I think, really maybe engaging more, aside from, using data, engaging more with people that are part of those subgroups to learn from, what's needed, what are their privacy concerns when feeding these types of data into any type of healthcare information, I think would be key to maybe consider. And I know we are not in an ideal world, but I think those could be steps that can be taking us in the right direction.
Thank you, Greg.
Yeah, I'm really glad you brought this up and mentioned it in the context of kind of rare diseases, and it's very much that same way in oncology. So we're talking about populations of folks when you've identified that they have one type of condition and live in this specific state, treated at an academic hospital, and have this mutation. All of the linking of different types of real world data make those types of privacy concerns all the more palpable, I would imagine, to patients. And then, for all researchers and folks involved, the methods that we are applying are becoming equally advanced. So things like tokenization in order to kind of identify patients and link these different sources of data together. I'd love to see progress that kind of makes this less of a barrier, because it really blocks things like understanding other types of patient subgroups that are often just othered. So like Asian Pacific Islanders, for example, it's a very heterogeneous group, and also Latinx patients. And so the concerns about kind of creating those small cell sizes, it really blocks us from kind of unveiling some of these possible inequities that happen in just general health in those groups. So I hope that, as these kind of technologies advance that kind of make the data linking possible, that privacy is kind of all part and parcel of that. And to Evelyn's point before, kind of securing that from the beginning and assuring patients that their data is protected.
For sure. And actually, we have an interesting question from an audience member along this vein. On the lines of data privacy, how can we continue to create linkages, even with omics data, using tokenization and other mechanisms, and share this data without bumping up on potential re-identification? Any thoughts from our panelists? And I know, Greg, you mentioned, data linkages and things like that.
A challenging question for sure. So thinking about something like tokenization and its role there, obviously, as again, linking these multiple sources of data together, it's going to become more and more of a challenge. So fully acknowledging that there, I think that, with respect to omics data and say like population level data, de-identifying those, absolutely a challenge. I think that there's processes like, for example, maybe not in the setting of omics, but say looking at area level or geographic level characteristics like census tract. There's different levels and de-identification processes that can allow you to include something that's not a full zip code or 9-digit zip code that would identify the patient, but use something else that would kind of identify within a specific geographic unit, and then that doesn't have to be part of what's revealed. But I think all of this together, raising more and more of a concern, especially as we think about this kind of precision real world evidence.
Thank you. I do, unless anyone has any additional thoughts, I have one final question before we wrap up. All right. Well, I want to talk a little bit about what was mentioned in Nora's presentation regarding differences in just terms that we use, whether we're saying effectiveness or efficacy, or a subpopulation versus a subgroup. So in a well-controlled clinical trial setting, conclusions can be drawn regarding a treatment's efficacy. In a randomized, controlled, gold standard clinical trial, that is possible. But in real-world settings, a treatment's effectiveness could differ from efficacy, especially regarding patient subgroups, or at least allowing for certain signal detection mechanisms to be used to identify subgroups that have a greater likelihood of responding to treatment in the real world. So how can we draw conclusions around effectiveness in different settings for different patient subgroups?
I think I want to go first. I see we unmuted at the same time. Okay, so I can take a first try at this one. So I think I sound like, you know, I'm repeating myself, but I think it really is we should consider you know, ways we can incorporate and learn without necessarily making it like a cookbook and everything follows the same recipe. I think it depends on the subgroups. It depends on the research question. And it really depends on what are the settings where the research question is best answered. And so you mentioned efficacy in clinical trials, and if the subgroups are not even considered there, and in my example with the cognitive you know, health and how people might respond differently to treatment, it's very relevant, right? So if you don't consider that some people inherently will respond slower or will maybe their impairment would evolve faster than in other groups, even though when you clinically evaluate them for inclusion, they look alike, right? Their test scores are similar, but just their disease progression is different. And there might be work that we still need to do in you know, trying to be able to identify these subgroups. So I think first, considering subgroups and ways to identify them to maybe signal detection in clinical trials that then will allow for additional real world studies that can tease out the heterogeneity would be important. But then with existing data and maybe in the more observational or pragmatic trials world, really thinking more broadly and trying to investigate additional subgroups that make sense for the research question, I think that is key. Again, I think I see every one of these as a piece of a puzzle, and the conclusion and the integration should be based on the evidence coming from different angles.
Absolutely. Thank you so much. Well, we are at time, unfortunately, and we do have to move on to session 3. So thank you, Greg, Daniela, Evelyn, for all of your remarks and for, you know, sharing your perspectives and your work. We certainly appreciated having you all here. Again, we encourage audience members to continue to submit questions to our panelists and presenters. There may be an opportunity for those questions to be answered at a later time, but for now, thank you again. We're going to go ahead and move on to session 3. I'm going to pass it now to Dr. Trevin Locke, who's our Assistant Research Director of Real World Evidence here at the Duke-Margolis Center, who will lead our session 3 discussion focused on generating practically relevant evidence in the real world. Trevin, I'll pass it to you.
Thanks so much, Rachel. Looking forward to a great session here. I've already heard many of the points that I'm about to talk about come up from our earlier panelists. So we'll get right into it. As Rachel said, I'm Trevin Locke, Assistant Research Director here at Duke-Margolis. I'm going to give this quick presentation and then hand it over to a fantastic panel. So I'll start by overviewing the last of our brand new white papers, this one on learning health systems, which pulls in a lot of the threads we've been hearing about all afternoon. And I'm going to tie that back to something we also heard about earlier today on leveraging real-world data and point of care and pragmatic trial settings, summarize that all up and hand it over to our panel. So next slide. And next slide. So I don't think I have to tell most of this audience that the concept of learning healthcare systems has been around for quite some time. The Institute of Medicine, now the National Academy of Medicine, has released several volumes on it, different aspects of implementing learning healthcare systems. AHRQ and others have picked up on that work to define key concepts and lay out learning health principles. But ultimately, and kind of the reason we honed in on this as some work for our review collaborative is that fit-for-purpose data is critical to fuel learning health approaches. As our ability to collect and analyze real-world data expands, so too do the opportunities to leverage that data to drive all the important work that we want to do and have been talking about all afternoon. Which leads me to the next slide. This has been recognized not just by us, and I'm sure many of you on the line here, but by leaders in the space, including leadership at the FDA. We heard a little bit from a couple of those folks earlier this afternoon, but I also wanted to call particular attention to a few pieces by current Commissioner Rob Califf, as well as former FDA leadership Amy Abernathy, all talking about the time for world data to become routine and really to fix our overall evidence generation system, while acknowledging some of the implementation and ethical challenges that integrating evidence generation in healthcare systems might present. So I'll continue on from there into the next slide. You can actually go ahead and go to the next slide too. So this all kind of led up to the white paper that I mentioned. As I said, learning health concepts are well known, but ultimately implementation varies a bit for some of the reasons we've heard today, as well as others. So we wanted to explore how real-world data is currently used in a cross-section of health systems to ultimately try to identify recommendations and considerations for further leveraging that data to advance our goal of learning healthcare. I think the link should be dropped into the chat for this paper as well, but I'll summarize some of our key takeaways here. Next slide. If you're at all plugged into the learning healthcare conversation, I'm sure you've seen plenty of iterative circle diagrams like this, but for those of you that haven't, the basic idea is that you take the data generated during the course of usual care, turn that systematically and intentionally into real-world evidence, use that evidence for various comparative assessments or other questions that you have in your system, and then alter your care decisions and approaches based on that evidence, and then start the process over again, continually iterating to improve care delivery and improve outcomes for patients and overall efficiency of the system. So next slide. So our work here, again, like the others we've heard about today, was originated from a 2022 RWE collaborative work stream focused on learning healthcare systems. Unlike the other two groups, we took a slightly different approach, working with our work stream to design a quantitative survey and some qualitative interviews with a range of experts within health systems across the US to hone in on kind of the topics that I've been highlighting. So I'll go through some of our key takeaways from that now. Next slide. And go ahead, next slide. So for the survey, I should say, the details on how we conducted both the survey and the interviews are in the white paper linked in the chat if you want to read a little bit more about who we were able to reach and how. But generally, we found, health systems in the United States are amassing substantial amounts of data, as we know, and using it to enhance critical functions such as patient care, financial management, operational efficiency, and quality enhancement. As might be expected, EHR and claims data stand out as the primary sources of that data due to their relatively widespread availability and the extensive structured data that they offer, at least when organized well. Survey findings indicate, though, that despite this accumulation of extensive information from various sources, the translation of that data into actionable insights and practical improvements is not happening as consistently as we might like. So next slide. So from there, we dove in a little bit deeper with some of our experts in the interview, and from those conversations came out these themes around the need for good metrics and utilizing them appropriately, building from good data sources, engagement and external collaborative partnerships to enhance the available data, selecting the right data source, and making sure it's fit for purpose. We've touched on that a little bit earlier today. Looking at devising incentives, both at the individual provider and system level to participate in this type of work. It's not necessarily an easy thing to do just because we think it's important. It does take effort and time. Also important related to that is fostering an organizational culture from the top down that this work is important. Likewise, the need for strategic long-term planning to make sure that the work we're looking for here is sustainable. And then finally, there's a need to address other internal and external barriers to implementation. So with these learnings, we developed on the next slide a list of challenges and some strategies to address them. So we've actually already heard about a couple of these from our fantastic panelists earlier in the afternoon. And this is not our complete list either. You please check out the paper for everything, but I'm highlighting a few points here that I think will lead to a great discussion with our panel. One, as was mentioned earlier, the administrative and time burden on providers here for helping collect this data is notable. So there's a need for sustainable uses of infrastructure and digital tools, including perhaps wearable devices to enable easier collection of data. But that's not enough on its own. We also need appropriate support staff and supporting tools to make it easy to collect and aggregate and analyze that data, managing provider burnout, and not putting this all on providers' shoulders when they're just trying to provide care to their patients. Likewise, we heard from several folks about a lack of needed expertise to make the most use of the data we are collecting, particularly in the informatics space. So, there's a need both early on to make sure training programs are incorporating the principles that we want to see in learning health systems and the appropriate training to execute those principles from developing students that will become our next generation of providers and support staff, but also offering opportunities for existing providers to kind of catch up on the latest and greatest in the space. Getting into the culture point a little bit, we talked with some folks about the challenge of low buy-in from both the patient and the provider perspective. On the patient side, I think the important drumbeat there is, patient centricity is important, and incorporating patient voices throughout kind of the development of both the core learning health system and any kind of research being conducted within it is important. And likewise, of course, making sure patients understand how the data is being collected, how it's being used, and for why, and who is overseeing it, and to the point that came up in the last conversation, making sure privacy protections are in place and appropriate. On the provider side, again, they are already quite burdened, so important to make sure that they see the value in participating in these learning health systems, whether through incentives for improved outcomes or just seeing the effect of these systems on outcomes and efficiencies in care pathways. And last, but certainly not least, data remains quite siloed within some health systems, and that lack of interoperability, even within certain health systems, obviously is a detriment to fully implementing learning health goals. So it's really important to work to make sure EHR is interoperable with each other within sites and kind of broader. And we'll talk, I think, a little bit more in the panel about standards and processes for maybe doing a little bit of that. And there's some work within the federal government from Office of the National Coordinator and others to help support some of this work. Next slide. And next slide. So here I'm going to switch gears a little bit. The concept of point of care trials was already introduced by our panelists earlier, so I'll run through this pretty quickly. But the ultimate goal is to or vision is that we should be able to conduct randomized clinical research in routine clinical practice settings and give patients and providers clinical research as a care option where they might not otherwise have it. And we should be able to do this in a way that is robust enough to inform decision-making by FDA, CMS, and other major decision makers. So again, if we can get the learning health development piece right, it should enable this type of integrated approach is kind of the vision. Next slide. You saw a slide like this from our keynote earlier. This is a little bit different take, but same basic idea of the randomized trial approach continuum. We kind of see point of care trials as kind of an extension of the vision of pragmatic trials, occurring where patients receive their routine care, integrating with EHR systems for core functions like enrollment and randomization, reaching patients where they are, where they get their care, providing an experience that they're comfortable and familiar with, and integrating those workflows to allow for streamlined data collection. This is not to say that they're the only approach and we should throw out randomized clinical trials. It's just one approach of many to helping us best leverage the data that we're collecting to generate practical evidence. Next slide. So how do we get there? We've heard a little bit already today, but there's some regulatory and policy issues to talk about. Likewise, there's some technology and data challenges, and again, it comes back to fostering a strong culture of research. And I'll touch on just a little bit of the work that we've done here at Margolis, both in the RWE Collaborative and within a separate coalition for advancing clinical trials at the point of care on just a subset of these issues. So next slide. In the policy space, we've seen a lot of activity recently, actually, from FDA and other policymakers. I'm just going to highlight a few of those and some outstanding questions here. So just this month, comment periods are closing on update to the International Council of Harmonization's Good Clinical Practice guidance that is now designed around kind of a principle first approach to good clinical practice built on risk proportionate quality by design approaches. It touches on investigator oversight and responsibilities for various stakeholders, including sponsors and investigators. One outstanding question that we've thought about here and in other policy efforts from FDA and elsewhere is, as we think about this new approach to clinical research and integrating research into care, what does an investigator look like? It's a bit of a different model than the traditional trial approach. So where are the lines between investigator, sub-investigator, and just general healthcare providers that are maybe collecting a piece of the data that will ultimately inform a research study? And so we are thinking about this, many others are thinking about this, but we've identified it as a key area to make sure we kind of get right to really integrate and achieve what our vision here for learning health and point of care trials. Likewise, we're seeing a lot of activity and interest really in building responsive trial capacity that's kind of warm-based and ready to go and for the future health emergencies. And we think point of care trials is a real option here, but no one can kind of build that on their own, and it needs to be, collective action and support from the whole community to kind of make sure we get the right capacity and space to answer the right kinds of questions. Next slide. On the data side, we've touched a little bit on the infrastructure needs already, but the key takeaway here is that we want to integrate research into care. That will require probably reconfiguring some existing tools or building a few new ones to enable that research in the care settings without overly burdening providers. As I mentioned before, there's some ongoing efforts by some government and other accelerators to help address some of these gaps to improve interoperability and technology supports to leverage data most effectively. Next slide. The Coalition of Point of Care Trials that I just mentioned earlier released a few weeks ago a letter from our membership highlighting some of the culture topic and the role of health systems in this. And so this is just briefly summarizing some of that, the importance of working at the community to engage equitably with patients and potential research participants, thinking about how to contact potential research participants to broaden awareness and access of research, and making sure there's capacity at the site level and the provider level to make research feasible, sustainable. And I'm running out of time, so next slide, and just right to the next. So a few key takeaways. So we still need more work to create a robust data infrastructure that's interoperable, prioritizes a culture that prioritizes research and learning, and ultimately incentives that encourage sustainability. We need federal agencies, policymakers, and other RWE stakeholders to kind of meet systems where they are, recognize their interest in improvement, and kind of all work together to make sure we build the system that we recognize that we need. As the use of real data within health systems expands, the opportunities to leverage that data for practical evidence generation do as well. And ultimately, as I said, we need to work with all stakeholders need to work together. And if they do, we have the opportunity to improve our evidence generation system, to drive more efficient drug development, improve care delivery, and ultimately, of course, make for better outcomes for our patients. And I think with that, I will close and hand it to our director here at Duke-Margolis, Mark McClellan, to lead off what should be a spectacular panel.
Thanks very much, Trevin. I'm Mark McClellan. I'm the director at Duke-Margolis. I appreciate Trevin covering a lot of ground relating to generating practically relevant evidence using real-world data and linking clinical practice to learning, healthcare practice to learning. Also want to let you know that I'm an independent director at Cigna Johnson & Johnson Alignment Healthcare. and want to also introduce our panel for this discussion this afternoon. Andanette Amaru is the Director of External Partnerships and Projects at UC Health Center for Data-Driven Insights and Innovation. Frederick Masudi is Chief Science Officer and Vice President of Clinical Research and Analytics for Ascension Healthcare. Chris Linzell is the Director of Data Science and Biostatistics for Duke Clinical Research Institute. He's also a professor and co-chief of biostatistics in the Department of Bioinformatics and Biostatistics at Duke University. And Sally Oken is the Executive Director for the Clinical Trials Transformation Initiative, which is a public-private partnership between Duke University and the U.S. Food and Drug Administration. And also glad to be joined by Hilary Marston, who's Chief Medical Officer of the FDA. The CMO is the primary clinical advisor to the commissioner, oversees the Office of Clinical Policy and Programs. As you heard earlier, addressing this issue of accelerated learning is a priority for the FDA, as it is for many federal agencies right now. So this couldn't be a more timely discussion and panel. We're going to start off with some brief opening comments from each of our participants, and then have some time for discussion. So as you've been doing all day today, please keep those questions and comments coming. We'll get to as many of them as we can. And with that, I'd like to turn to Andanette to start us off with his opening comments.
Thank you, Mark, and thank you for the Duke-Margolis team for having such a wonderful conference. Delighted to be here with you all today. My name is Andanette Amaru. I serve as the Director of External Partnerships and Projects at University of California. And I want to, I'd like to share some insights on how, as University of California, we're evolving as a learning healthcare system. And before we dive in, I do want to note that I have no conflict of interest, and the views that I express today are solely my own and do not represent any official stance of University of California. Next slide, please. So the University of California Health is one of the leading health system and stands about 7th largest by revenue. We do have quite 20 health professional schools, 6 academic schools, and 12 hospitals, and our reach are pretty profound and impactful. UC Health is University of California. I think the slides are different here that are showing. I'll keep going. So the University of California is also a hub for innovation. We average 5 inventions daily. We've started over 1,500 or birthed over 1,500 companies based on University of California intellectual property through the end of 2022. With active US and foreign patents, we're showcasing our commitment to advancing healthcare in general. Next slide, please. Perfect, thank you. UC Health has a center for data warehouse, which is really, to me, the heart of the UC Health as a learning system, which is how we thrive. And our secure central data warehouse amalgamates data across all of our 6 academic medical schools and our systems. Our data is reflective of the diversity of California, which is the most diverse state in the US. It helps us facilitate operational improvement as well as quality patient care. It paves the way for cutting edge clinical research. This data is de-identified for clinical research, and it adheres to the most strictest guidelines and reviews. It is stored in an OMOP common data model that ensures consistency with other state national efforts. Next slide, please. And this data warehouse, again, is very rich repository of diverse patient data. Holds data on nearly 8 million patients that have been treated within our system since 2012. Over 810 million procedures, over 3 billion vital signs and measurements and test results, 600,000 primary care patients alone. Quite a bit of data, which is rich for our researchers. Next slide, please. And then as a learning system, we're continually evolving and adding more to the data landscape. Currently, our data is centralized in a Microsoft Azure Databricks secure cloud environment. Again, enabling collaboration among our community of 400 researchers who are driving the future of healthcare. Thank you.
Thanks very much for those opening comments, London. I appreciate it. Next, I'd like to turn to Fred.
Hi, Mark, and thanks to you and the Margolis Center for hosting this terrific meeting today. I've learned a lot, and it's a pleasure to be involved. So as you said, Mark, I'm the Chief Science Officer and Vice President of Research and Analytics at Ascension. We're a large health system, about 140 or so hospitals. Mark, can you hear me okay? Oh, you can?
Absolutely, just fine, yeah.
Okay, perfect. Yeah, we're about 140 hospitals in the central US, in the south and southeast, as well as on the eastern seaboard. You know, I started at Ascension a couple of years ago with the role of developing our research strategy, our national research strategy and program to build upon the research that was being done at Ascension in its multiple sites in a more federated manner. And we span the spectrum of clinical research, from the traditional clinical trials to implementation research, pragmatic clinical trials, all the way to data analytics. You know, with respect to our data analytics piece, we obviously employ real world data, not just for the purposes of data analysis, but also to support our work in clinical trials. You know, one of the issues we deal with in a health system, of course, is that in general, understandably, health systems don't typically view evidence generation as their key mission. And this is particularly so in the face of challenging financial times, which has been the case to an unprecedented extent in the last couple of years. And that creates a couple of imperatives for us. One of which is to ensure that the evidence generation activities that we are engaged in are well aligned with the mission and clinical imperatives of the organization. And then we can educate our leaders as to how this better aligned evidence generation process really is part of our key mission. The other is to foster an understanding of how a robust real world data, real world evidence program can directly support the organization beyond what might be considered research per se, so clinical operations, quality, and safety. You know, we are a very large health system. As I said, we don't have quite the benefits of the UC health system that were just described. We have multiple EHRs in a data warehouse that is cloud hosted, but as you can imagine, with a multiplicity of EHRs, that creates some substantial issues with respect to data normalization. And we can discuss, I'm sure, a lot of these issues in terms of barriers and opportunities as we proceed. So again, thanks for inviting me to participate.
Great, really appreciate your opening comments. I do want to come back to those issues around data standardization and usability, and around support for healthcare organizations to participate. Now let me turn to Chris Linzell for some opening comments.
So thank you very much for the invitation, and again, the wonderful afternoon of information that has been quite insightful. I'll place my comments in the context of a learning health system that has two components. The first component is learning from what we do, and the second component is doing what we learn. We spent a lot of time today talking about the first, learning from what we do, and talking about the real world data. And a big part of what we do is try to convert the real world data into real world evidence that is actionable. And so the data and the evidence are not necessarily the same thing. And in going from data to evidence, we have to think about the many things that have come up today, the issues around data quality, the issues around bias, the issues around missingness, and the issue around coding. Some of the areas that really worry me as we learn about what we are doing and what works for which patients is our ability to know about outcomes. Recognizing that if we have a single system data set and access to some claims data, we're likely missing a large amount of the outcomes that matter to patients and that matter to us. And so really trying to think about the outcome component of our real world data, and how do we ensure that we reach beyond the EHR and we reach beyond the claims data. And this is particularly critical because, as has been said many times today, the way that those data are generated is through a coding mechanism. And that coding mechanism is often human-based and is not necessarily based upon understanding the clinical outcome from the participant. There are other reasons for those codings. So moving beyond the idea that we really need to be able to access outcomes, the idea that those outcomes are needed in a sensible time frame becomes important. For many of us who are doing learning health system type work and learning from real world evidence, we need, for example, the fact of death. And yet the availability of the fact of death can be challenged by a couple of years. And it doesn't help me to run a clinical trial and to know that I've got 30-day outcomes, but I'm not able to access them until 2 years after the fact. And so just in the learning from what we do, I really think a lot about the data quality, the missingness, particularly around the outcomes, the temporality of the data around the outcomes. Now, moving to the second phase, the doing what we've learned. A lot of the times now we are using algorithmically informed clinical decision support. Those algorithms are based upon the real world data that exist. And now real world data are becoming the interventions that we are testing in our learning health systems. And this is an area where we need to pay extra careful attention to the issues around data quality, bias, missingness, and outcome assessment. Because if we are using the real world data to create real world interventions, we need to make sure that they are not resulting in misunderstanding in the real world evidence. So again, learning health system is learning from what we do, and we need to understand how the data are generated, and we need to make sure they are timely and correct and complete. But then when we start learning from what we do and using that information and applying it to clinical decisions, we need to understand how any issues that arose in the real world evidence translate back to real world care. And that is something that worries me on a day-to-day basis. On a positive side, we have people who are worried about it and are looking to fix it. Thank you.
All right, well, let's come back to some of those fixes in just a few minutes. But thanks for the comments, Chris. Sally?
Sorry, unmute myself there. It should be practiced at this by now. Thanks so much for having me here today. It's been a wonderful day. The Clinical Trials Transformation Initiative, as Mark mentioned, is a public-private partnership of the FDA, and we're housed at Duke University. We're also a member organization that seeks to develop and drive the adoption of practices that increase the quality and efficiency of clinical trials. We do this through a multi-stakeholder approach, and creating consensus-driven, evidence-based products is really the hallmark of our work. In 2020, Citi launched our Transforming Trials vision that includes 5 pillars and describes how clinical trials should be done by 2030 as a critical part of an evidence generation system. These include, briefly, Pillar 1, clinical trials be patient-centric and easily accessible. 2, that they be fully integrated into health processes. 3, that they be designed with a quality approach. 4, that they maximally leverage all available data. And that they ultimately, for Pillar 5, improve population health. With all 5 pillars, they really are applicable to advancing an evidence generation system within a learning health system. I want to focus on Pillars 2, fully integrating health into health processes, and Pillar 4, maximally leveraging all available data, because they really are especially relevant to today's discussion. Citi has several recommendations, resources, and tools related to the various elements of embedding clinical trials into clinical practice that align with the learning health system principles. We're also actively working with groups such as ACT at POC to identify and participate in opportunities for applying these evidence-based recommendations and tools into pilots as well as into practice. I'd like to focus my remarks on the importance of fostering a culture of participation that Trevon mentioned earlier. Specifically, the participation of patients, potential participants in a clinical trial at the point of care, and their providers. As we seek to integrate research and care into a learning system, it's going to be essential that we intentionally involve each of these groups in trial design, in the processes used for the conduct of the trial itself, and importantly, in sharing new knowledge and learnings that come from that participation. One important example is to include patients and potential participants in evaluating opportunities to modernize our informed consent process. For example, when a waiver is granted for a study that will be conducted within clinical practice based on the determination of minimal risk, ought we ensure that these processes are in place for patients, participants, and their providers to have an opportunity to be part of that risk assessment, that risk determination. Now, if we consider Pillar 4, which seeks to maximally leverage all available data, we know that certainly this will be important if we're going to achieve the promise of a learning health system. Yet the implications for privacy, confidentiality, and safety loom large, particularly given the rapid emergence and integration of AI and ML. So brings me back to informed consent again. For those studies within clinical practice for which a waiver of consent isn't appropriate, can we better engage patients, potential participants, and their providers in the evaluation and analysis of models of consent that, in essence, seek to give patients greater control of their options for participating in research at the point of care? Examples such as broad consent, which asks research participants to consent to multiple future studies, the nature and specificities of which are not quite known at the time, may be problematic, but there may be elements of it that we should be considering as we modernize. The idea of a dynamic consent is a process that seeks to continuously engage participants based on their personalized communications platform and facilitates a two-way communication between research and participants. These models are not new and have been used in biomedical research for some time. Might it be time to truly challenge ourselves and consider how we can use models such as these and modernize them with patient, participant, and provider-centric technological capabilities to ensure transparency, personalized controls, and the support of a continuous and shared learning environment when we are doing the integration of research into care? So thank you so much, and I'll leave it there.
Thanks, Sally. Really appreciate those comments. And I'd like to turn now to Hillary Marston.
Thank you so much, and thanks for the invitation to join this panel. Obviously, coming last in a long series of expert presentations, I'll see what more that there is to say. So Trevin, in his opening presentation, which was really excellent, noted that the commitment to the learning health system is not new. I'll also say that that date back to 2007 is one that our current commissioner shared in. He's been part of these discussions and really committed to pushing this forward from its inception. And the promise for RWD and RWE studies in the learning health system is clear, but I also want to focus on the promise for improving evidence-based practice. So first on the RWD, RWE type studies, which are more in our purview at the FDA, you've already heard a great deal today about the work that we're doing in this area. You've heard about the framework that was put out back a couple of years ago. It outlined the guidances that we intended to release to the sponsor community, and we've been executing against that. So those are coming out on a regular basis. A couple of those are still in draft form. Donna then eloquently walked you through some of the examples of how we're actually using this in practice. And as we look forward, we're going to be incorporating the lessons from those approvals, from things that are successful, but we'll also be incorporating some of the lessons from the grant funding that we're doing. So what are we actually learning about our ability to use these data sources? All of this will be useful as entities like UCSF and other medical centers that are committed to produce into the learning health system try to put them into practice. Turning to the second type of study, though, evidence-based practice, I think this is an area where we as a public health community, as a research community, have quite a bit more work to do. This is much of what we're doing in clinical practice, we're all aware of this, is not evidence-based. As you look through the guidelines for cardiology, oncology, et cetera, much of what we have requires more evidence, as noted in the guidelines. So what can we do? Committing to the learning health system is an important part of this, and I think we've seen progress in that area. I think also there's probably an element of the learning health system being really intimidating. It looks, as it's framed, like something that's all or nothing. Like we need to commit our entire health system to it, and probably is the purview of institutions like Duke, like UCSF. I think breaking it down into manageable chunks and what can be used in community settings is a useful part of this conversation. What can we do from a policymaker point of view? We're going to get into some of this, but there are a couple of levers that we have at our disposal. So certainly work to be done on data quality and standards. You've already heard about a good deal of that. We can look at some of our reimbursement practices. So that's certainly not the purview of FDA, but needing to make sure that the way that we're reimbursing clinical care doesn't actually get in the way of these research activities. And then from a leadership point of view, conversations like today are critically important. I also think that getting the payers more involved in these conversations is really key here. They need to be able to see the value proposition, what they can actually get out of it, what will help their bottom line from these types of activities. Currently, the health insurance providers have been somewhat outside of the conversation, and I think we really need to draw them in. So thanks for the opportunity for a few comments there. Look forward to the discussion.
Thanks, Hillary. Thanks all of you for some really thoughtful comments to get this discussion going. As you said, I wish we had more time, but I do want to highlight and follow up on some of the issues that you raised. And maybe we could start with something that I think all of you emphasize in one way or another, which is the importance of having data. As going back to Kara's opening remarks about fit for purpose data, Chris noted that data may be incomplete, may be driven by coding practices, which in turn depend on things like reimbursement. We heard from Fred about differences in the way data seem to be realized across different EHRs and different healthcare organizations. What are the next steps that are needed to make progress on the data front? Is this a policy issue? Is this something that continued progress on data standards and digital tools is gonna help improve? Any of you want to comment on that?
Mark, I'd say it's probably all of the above. Like it's a lot of those things at work. I think that obviously with the adoption of EHRs, I think there was a big focus on getting them into play. The EHR vendors have focused a lot on making them very efficient billing machines. And so as a result, they're very interoperable when it comes to billing and not interoperable with respect to those things that matter to clinicians and patients. And then I think they allowed sites a lot of flexibility so that their clinicians wouldn't revolt when they were using the platforms. And as a result, clinical interoperability really suffered. It's a big challenge, even within a single EHR and an organization like ours that has dozens, literally, of EHRs to create that kind of standardization. And if you're not going to pay for the front end cost of adopting a single EHR in a large health system, which is a 9-figure investment, you have to do a lot of back end work that's a never ending battle. And to Chris's point too, we're not necessarily collecting the EHR everything that's important to our patients and to us. So in our case, we don't necessarily have a line of sight to all the healthcare activities of individuals who see us in our system. And importantly as well, we aren't necessarily capturing patient-centered outcomes. Now we're doing that in specific cases, in orthopedics, for instance, doing a very good job of capturing patient-reported outcomes for registries. It's been motivated by some incentives and some policy incentives. So my belief is it's probably all the above to move forward on this, but obviously a huge barrier to generating the kind of evidence we need to be a true learning healthcare system.
Thanks, Fred.
I'd like to pick up, if possible, on something Hillary mentioned and ties into Fred's response, and that is that I think that the payers and the financial drivers behind the systems need to come to the table. And we need to be able to address the different priorities in how we code up our data and how we create the structured information. And we need to recognize that we're at a time when it's important that the money flows right. but we also need the clinical information to be correct as it flows. And then just to build upon that, I fear that we may be in a disruptive moment when the way that we document clinical care is going to change on the backs of things like generative AI. And we're beginning to see the transcription of notes that are taken by clinicians at the bedside being coded by AI. So this is both a fear and an incredible opportunity. If we can get that bit right, then we can create structured data from which we can actually learn very, very efficiently.
I appreciate those comments. And Anya, maybe I can bring you in here. You've had some experience in integrating data across a number of different organizations across the University of California. Any thoughts you'd like to add? And then I want to move on to how we accelerate the progress and address some of the concerns that Chris just raised.
I absolutely agree both with Fred and Chris. I don't think we can wait for the policy side or the incentive side, but I do think things need to be done in parallel where we bring in the payers and additional partners. And again, I think even on the sides of generative AI, I think we're doing a lot in terms of trying to understand today, as we sit here, how do we supplement the data that we already have by analyzing notes, right? There's a lot of information that's buried in there. So I think it's an all of the above approach. There isn't a cookie cutter approach that's done, but also having these types of platforms to be able to share with one another. Hey, here's what's working, and it may look very different as what's going on across the different care facilities, but sharing what's working and then understanding how we can lift one another is going to be the future.
Yeah, and maybe Hillary, I could come back to you and Sally on this, and like sort of what some underlying sources of truth as we move into an era of AI and richer sources of data as well. You mentioned, Hillary, about starting somewhere, that not maybe going to trying to standardize every possible data element in different areas of clinical care where we aren't seeing as much evidence guiding practice as we'd like and seems like might be possible today. Are there sort of a starter set of key data elements like patient outcomes or important comorbidities, or what's the right way to frame the progress needed around steps, as Ananette was emphasizing, that organizations can take, but that also could be reinforced by policies, especially policies that are now focusing more on improving outcomes?
Yeah, so I think this question is critically important because a lot of the answer comes from where you sit, right? So what you're going to prioritize is going to be heavily biased by what you think is most important to study in the health system. Right now, there's a good deal of momentum behind the Cancer Moonshot. You heard about that from Donna Rivera. So they're looking at structured data elements that would be critical for cancer trials. You could certainly see a similar effort for cardiovascular trials. I think what we're going to see there is that there will be a good deal of crossover behind those within those structured data elements. And I think we'll also see that investigators are going to look at the structured data elements and the other ones that they wish that they could collect and probably peel down some of what their wish list would be. I think quality by design work from Citi will be essential to that sort of work. But you're not going to be able to get everything out of the structured record. AI, this is something that we're enthusiastically looking at. We put out a discussion paper about uses of AI in drug development, and going all the way from basic drug discovery through to these sorts of studies. We definitely think that there is good potential for AI to help organize the data, clean it, analyze the sources, probably also help in some of the protocol design. But we're still going to need to maintain our standards in terms of the way that the data is presented to us and the ability to trace back the decisions that are made. So the black box problem is one that we're concerned about and something that's going to need to be addressed for us. Thanks.
Yeah, just I'll sort of channel some of my own payer background, formerly at CMS and all, is that payment is moving in the direction of some of the outcomes that you described too, not just traditional, fee-for-service coding. So for example, in Medicare now, you mentioned cardiovascular trials and evidence having accurate data there, things like blood pressure control, lipid control, hemoglobin A1Cs are being used in payment and performance reporting. So maybe some further opportunities for alignment there with steps like the Cancer Moonshot and other research initiatives. Sally, Citi got called out by Hillary for what are good practices here, and you emphasized a lot of patient perspectives and patient engagement, getting better data in your comments. Anything you'd like to add?
Yeah, I mean, I think one of the key aspects of the work that we do is the multi-stakeholder aspect of it. So we are constantly looking to ensure that we've gotten the perspectives as needed for any consensus-driven evidence-based project we're working on. And certainly our quality by design work definitely incorporates that sort of approach. And it is an area that we're working really hard with FDA to sort out how can we begin to really actively implement some of the practices and tools and resources that we know are really great to improve processes, but ultimately do require that foundation of trust and a culture of participation when you start getting into the clinical trials in the point of care environment. And so I think those are going to be opportunities for us to really test our assumptions. The other piece I would just say is that we have constantly said all along that there is no one size fits all on this. And it almost crosses over all of our recommendations. And it's a bit of a trite statement, but it's just so true. And so I think what Hillary said was, your answers are going to come from where you're sitting in this system. And where you're sitting in this system will oftentimes then influence how you're going to react to any particular innovation or something that comes down the pike. I will say lastly, my work with patients over the many years has really shown me that when we talk with them about the opportunities for learning and then ensuring that we will share those learnings back to them, so many people believe that that should be how our system works. And frankly, many patients have said to me, what do you mean you're not learning from the information that you're gathering from me on a regular basis? So I think we need to step back and say that's a particular perspective that we should be building on in terms of that culture of participation, because without it, we're not going to have the participation that we need to really ensure that we have that data that we know is going to be so rich to really have the outcomes we want for patients and providers to have decision-making capacity. So I'll leave it there.
All right, I want to turn to learning next, but just to get to comment that came in that we've already touched on a bit. Relates to data ownership and potentially leading to fragmentation, lack of sharing, lack of comparability, some of the issues that you've talked about. Are there any further policy steps that you think are needed to advance progress in consistent, reliable data that can track patients over time? Anything else that this lightning round on this topic that you'd like to add to the discussion?
I would simply add that it's hard, but not impossible. So we should have the technological capabilities and the intellectual capacity to be able to solve it, but we have to solve it with the participation of those who are going to be most affected by it.
I think my comment would be that the data pretty much exist, but we don't know how to get it. And our systems and society and culture prevent us from efficiently accessing what already exists. And so any policy changes that can help us overcome some of our individual barriers to sharing information might help us move towards even just sharing death data in a rapid way.
Yeah, and it does seem like there is some growing potential for alignment between goals of agencies like FDA and ARPA-H, Cancer Moonshot, with advancing better longitudinal, reliable data as a basis for learning, interests at CMS around promoting a focus on better outcomes and greater equity, and other payers as well, and interest within the healthcare organizations themselves and making a difference for their patients. Wish we had more time to discuss that. I do want to talk next about what to do with the data in the remaining time that we have, and we've talked about today on several different iterations a range of real world evidence generation methods, as Chris was saying, going from the data to the learnings and the actionable evidence that included observational approaches all the way over to traditional clinical trials, and maybe a growing space in between with more flexible or limited focus on data, not quite as comprehensive data collection, maybe a simpler randomization, pragmatic approaches, point of care approaches with more limited data collection. So there's a spectrum there. Chris, since you brought up the topic of data not being the same as learning, maybe we could say a little bit more about how we can advance both the reliable use of observational real world data studies to advance learning, and when randomization might be needed.
It's a really very good question, and perhaps one that's almost impossible to answer. Ultimately, any data, whether it is a perfect randomized controlled trial or whether it's a terrible observational study, every data element has information content that is both correct and incorrect. And what we need to do as scientists and researchers is be able to differentiate when that information content is directionally correct and when it is biased, inappropriate, and fraught with misinterpretation issues. And so as we move from the real world evidence space into the randomized controlled trial space, the certainty with which the information content exists moves from being uncertain to more certain. And I think as long as we respect that and we bring it into the decision-making and the analysis components, that's a really beneficial thing. I do worry a lot about people creating synthetic data. It could be highly effective. However, the assumptions creating that synthetic data from which to draw inferences really drive those inferences. And if we knew what was going on, we really wouldn't need the synthetic data. And so I'd say we need to respect the information content. We need to understand its uncertainty. We need to actually recognize a computed phenotype as probabilistic, not deterministic. We need to carry that into our analyses. And we need to stop thinking that we can make things up to fill in the gaps until we understand how we're making it up. And then I'll be okay with it.
So given those concerns, maybe Fred, and on that, I'll turn to you for the follow-up, which is, okay, that sounds like it could be a lot. On the other hand, there's an increasing amount of, as Chris was saying, valuable information that could be used to help guide decision making. What do you all advise as kind of next steps here? Where are you focusing your own efforts to build out relevant decision relevant evidence for practice and maybe for policies and other population health steps too?
You know, what we're doing is embedding ourselves in the clinical enterprise. So we're working very closely with the service lines, with the clinicians and clinical leaders to define what their, what the most important questions are to them. And then our role is to be enablers of generating the evidence that they feel is the highest priority. And that's the key, I think, the difference between the position I'm in now versus where I was in an academic medical center, is this idea of embeddedness is absolutely critical, and taking the lead from the frontline clinicians and the people who are really facing the problems, and helping them think about how best to address them, be that through an interventional approach versus a data analytic approach is the way we've chosen to deal with this and to prioritize.
I think Fred hit on a key word, enablement, right? I think that's exactly what our team is doing, is enabling our clinicians. We've got a few models that's worked for us with clinical collaboratives where folks across the 6 academic health centers are reviewing data, right? And I will tell you, it's not perfect data. A lot of the times they'll say, go back and look at this, or go back and pull this for me, right? And then we will establish a dashboard from which they're able to go back and figure out an anomaly or why certain treatments are resulting in different outcomes, right? And from there, they'll start to create a best method that's shared across our systems. That has a resounding amount of success, and that's allowed us to continue to do what we do at our Center for Data-Driven Insights and Innovation team. That's one, and I think we're continuing to progress within that. We're creating new molecular dashboard across our 5 cancer centers where folks are looking at, again, these molecular dashboard and cases being presented. So it's really benefiting the patients, but we're just starting to scratch the surface with what we've created at UC. But I will say the future is exciting.
Yeah, it sounds like you've created this virtuous cycle of, by looking closely at relevant questions, being able to identify underlying issues with the data that may need to be addressed. And in turn, that gives you more confidence, back to Chris's point, about the evidence that you're generating, and hopefully in turn can provide a stronger foundation for using the same data and same infrastructure for additional studies. I want to go back to Sally, since you raised a number of issues related to patient participation in all of these efforts. Anything that you'd like to add before we wrap up on the on next practical steps to get patients more engaged and embedded, since that seems to be the key process here for clinicians?
I'd be really interested to hear some of Andelet's and sort of Frederick's experiences of doing this at the point of care. But I do think we need to get the stories. We need to hear from patients and understand what their concerns are in the idea of having research being done as part of their care, or having research be a care option. I don't think we have enough insight into that. I don't think we've asked enough questions. And I think the opportunity to learn from that would actually help us be able to drive where we need to go. But without it, we really are sort of heading in a direction that ultimately patients may balk at and not want to participate in at all. So lastly, I'll just say every bit of data represents a part of a person's story, and we have to absolutely respect that and figure out ways of ensuring that it can be used ultimately for really important, efficient, and effective decision-making.
Yeah,
for them or someone else, frankly.
Improving care for them or patients like them, it's in their whole care journey. Well, I wish we had more time for all of this. Hillary, I'd like to give you just a last minute. Any final comments you'd like to add on FDA's perspective on how to accelerate progress and move forward from what seems like a foundation that's getting stronger, but still a long way to go?
Yeah, quite a responsibility to wrap up here. And I hope that you will actually wrap up after me. So I think we set out our views on where we were going to go in RWE and RWD. And we have been executing against that. Where this meets the learning health system is pretty clear for the studies that we regulate. Where we run into issues is where we're beyond what we regulate, the post-approval setting. And I think that's where the conversation really needs to be here. We have a good deal of work to do here. I think, and I mean the broader public health community. I think we also need to recognize that for a lot of the patients that we're working with here, it might be their first experience in clinical research. So to have them jump right into a system where everything that they're doing is potentially open to a research question is a lot. So we need to think about that as we're approaching patients and doing true informed consent with them. But there's a lot of potential here. We are just skimming the surface of it, and I think we're going to continue to learn quite a bit in coming years. So look forward to engaging in this next year and seeing how far we've come. Thanks.
Great. Thanks, Hillary, and thanks to all of our speakers and panelists in this panel and throughout the day who have shared their insights with us. We certainly covered a lot of ground. I just would begin my wrap up by saying that focus on the post-approval setting after those pivotal randomized controlled trials that are far over, I guess, at the left side of that chart that we showed earlier on evidence generation, where those conditions aren't as well suited to what's feasible and useful to learn for guiding decisions for physicians and other clinicians and patients and individual practices. That is an area where the Real World Evidence Collaborative we've talked about today is obviously continuing to do more work. It's also an area of activity for our Advancing Clinical Trials at the Point of Care Coalition, range of collaborative activities with the Clinical Trials Transformation Initiative and the Duke Clinical Research Institute. So please stay tuned, and we look forward to continued collaborations and progress on all of these issues. And just to briefly summarize what we talked about today, which I cannot do justice to in just a couple of minutes, we heard at the beginning from our colleagues at FDA about just how much activity is taking place there in terms of new guidance documents, demonstration projects, research projects that the agency is undertaken to engage the broader community around advancing evidence development and increased participation in those efforts too, recognizing this is not just an FDA issue, but involves other government agencies. We've talked about a number of them in this session. We also heard about the importance of leveraging and building broader experience and capacity for effectively developing, understanding, and then using on a fit-for-purpose way data that is generated in routine care with support from protocols and attention to appropriate rigor for the purposes of those data uses and their therefore their contribution to effective learning, actionable, unbiased evidence development. And we talked about the importance of diversity and inclusion and the importance of clear protocols to support consistency, rigor, efficiency, bias mitigation, and balancing diversity and inclusion with privacy, and the very important issue of earning trust. Engagement came up again on the last panel, both in terms of clinicians who are very busy and have other things that they have to do, but really want to improve care for their patients, and the patients themselves, as Sally very eloquently was saying, are on what's often a difficult and uncertain and costly journey from the standpoint of their health, the health of their loved ones, and want to get a better result as well. So lots of good ideas to address these big challenges, but also equally big opportunities to improve evidence development. And I really appreciate all of you being here for part of this. I have to give a special thanks to our team at Duke-Margolis, Rachel Hendrick-Sturrup, who served as moderator, Nora Emmett, Trevin Locke, who presented, Matt D'Ambrosio, Sandra Yanka, Yuk Durocher, and Mariam Nafi, who assisted in the planning and logistics for this meeting, which is a lot of planning and logistics. And a thanks to the members of our Real World Evidence Collaborative, many of whom have been engaged with the center on these topics, as we've seen an evolution from not having clear frameworks to having some early progress to really having a lot of activity and momentum around real world data generation and validation and real world evidence, as we talked about today. So much more to come, but thank you all for being part of this effort, and I hope you have a good rest of the afternoon, and will stay in touch with us about these continued important activities around real world evidence for improving patient care and the health of Americans and people around the world. Have a good rest of the day.